Excellencies, distinguished guests, colleagues, and friends, good morning. Welcome to the 2025 Eubiologics-IVI Global Vaccine Forum. My name is Ari Im. I am the Global Communications and Advocacy Manager at IVI, the International Vaccine Institute, headquartered here in Seoul. I'm honored to serve as your moderator for today's program. We have a remarkable lineup of speakers to address novel vaccine technologies and innovations. The lectures we'll hear today are united by the theme, Present and Future of mRNA and Other Vaccine Platform Technologies. Together, we'll consider opportunities for collaborative vaccine development. To open the program, I'd like to invite to the stage Dr. Dokeun Kim, Director General of IVI.
Thank you, Ari. 안녕하십니까. Good morning to everyone in the audience. I'm Dokeun Kim, the Director General of IVI. Dr. Youngmi Ji, Commissioner of the Korea Disease Control and Prevention Agency, Dr. Young-ok Baek, CEO of Eubiologics, Excellencies, esteemed speakers, distinguished guests, and colleagues. On behalf of the International Vaccine Institute, it's my honor to welcome you all to the 2025 Eubiologics-IVI Global Vaccine Forum. This gathering brings together leaders, innovators, and pioneers dedicated to advancing next-generation vaccine technologies to protect and promote global health. It offers a unique opportunity to engage with distinguished experts from around the world, exchange knowledge and insights, and foster collaboration to drive discovery of innovative health solutions. Today, scientific breakthroughs in mRNA and novel vaccine platforms are revolutionizing how we prepare for and respond to future health challenges. The COVID-19 pandemic highlighted the critical importance of vaccines in controlling infectious diseases and saving lives, demonstrating that rapid innovation, strong partnerships, and scientific expertise are essential for global health security. mRNA and next-generation vaccine technologies hold unprecedented potential, not just for rapid response to emerging diseases and future outbreaks, but also for targeting cancers, autoimmune conditions, and diseases previously thought to be untreatable. These flexible, scalable platforms enable faster development, broader immunity, and tailored responses. Throughout today's discussions, we will explore key topics such as self-amplifying mRNA platforms, cutting-edge vaccine technologies, correlates of protection, and strategies for overcoming challenges in vaccine development. These conversations will help us accelerate innovations that will shape the future of vaccines and immunization. I'd like to express my deepest gratitude to our distinguished speakers, chairs, and participants. Their expertise and commitment are essential to driving transformative innovation and ensuring that life-saving vaccines are made available and accessible to those who need them most. The insights and contributions shared today will not only propel scientific progress, but will help influence vaccine and health policies, both in Korea and globally, while strengthening our collective ability to respond to future pandemic health threats. IVI is the only international organization devoted to the development of vaccines for global health. At IVI, we firmly believe that collaboration drives progress, as evidenced by the development of the low-cost oral cholera vaccine from Eubiologics, which is gratefully sponsoring this forum, and a novel typhoid conjugate vaccine. These vaccines were developed by IVI and tech transferred to industry partners to achieve WHO pre-qualification. By bringing together scientists, funders, policymakers, industry, and research partners, IVI fosters a collaborative ecosystem for the discovery, development, and delivery of vaccines for global health. This meeting, which marks the 6th in our Vaccine Forum series, should inspire new research, create new partnerships, and accelerate the development of life-saving technologies. The collective expertise and insights in this room are transformational, working together, thinking together, creating together. Recent cuts in R&D and global health funding at a time when innovation and global cooperation are more vital than ever, and when it is important to defend the progress made in saving lives and improving the quality of lives in the past 2 decades. These setbacks will impede scientific progress. weaken our collective ability to respond to pressing health needs and future crises, and the effect will be generational. IVI, though, remains committed to its mission of discovery, development, and delivery of vaccines for global health and the empowerment of people through vaccine security. This forum is a timely opportunity to reaffirm our commitment to innovation, progress, collaboration, and impact through science. I'd like to take this opportunity to thank Eubiologics, Seoul Cyber University, and the Korea Support Committee for IVI for sponsoring this and the past 5 global vaccine forums. I'd also like to thank the KDCA for co-hosting this important event, and with that, I'll introduce Commissioner Young-Mi Ji.
Good morning, colleagues, distinguished guests, ladies and gentlemen. I'm Young-Mi Ji, Commissioner of the Korea Disease Control and Prevention Agency. It is my great privilege to welcome you all to the 2025 Global Vaccine Forum, co-hosted with the International Vaccine Institute and Eubiologics, where we gather together to discuss vaccine development strategies with a focus on mRNA vaccines and vaccine platform technologies. I'd like to extend my sincere gratitude to Dr. Dokeun Kim, Director General of IVI, and all partners who have worked hard to organize this Global Vaccine Forum. Also, I'd like to express my deepest appreciation to our chairs and speakers, especially those who traveled a long distance to participate in this forum, and everyone for joining this significant event today. With continued threat of new pandemics, KDCA established the mid to long-term strategic plan against emerging infectious diseases in 2023. As one pillar of this plan, we are making full efforts for securing vaccines and therapeutics within 100 or 200 days of new pandemics and epidemics. Securing cutting-edge vaccine platform technologies, including mRNA vaccines, is a key priority in preparing for future pandemics. In line with these efforts, KDCA launched mRNA vaccine development project this year to secure Korea's mRNA vaccine platform technology and commercialized COVID-19 mRNA vaccine by 2028. The Vaccine Research and Development Center and KDCA serves as national control tower to accelerate vaccine development in response to emerging infections and priority infectious diseases, as well as bioterrorism, and also to support research and development in securing new technologies in the field of mRNA vaccines. The Vaccine Research and Development Center has maintained a very close partnership with the IVI for past 10 years, especially during COVID-19 pandemic, for developing our own COVID-19 vaccine. Moreover, under the Korea Advanced Center for Vaccine Development COVADS, which we established recently in Andong, we aim to strengthen our national vaccine development capabilities. The main project of this center will be constructing vaccine antigen library through advanced technologies such as artificial intelligence. This center will also closely collaborate with IVI's Andong branch. As no country can prepare for and respond to pandemics alone, KDCA is actively collaborating with national and international partners, including UK Health Security Agency, US CDC, European CDC, Singapore CDC, and WHO, CEPI, and IVI. Especially, KDCA is also closely working with CEPI through CEPI's centralized laboratory network and 100 days mission project. IVI will be also one of the KDCA's valuable partners in this project. So I do hope today's forum will provide another valuable opportunity to discuss strategies for advanced vaccine platform development and to share and advance insight from government, academia, international organizations, business partners, and all experts. Once again, I thank you all for joining this Global Vaccine Forum and wish you all very successful and productive meeting. Thank you.
Thank you, Dr. G, for your support and inspiring words. We're truly honored to have experts and leaders from around the world with us today, ready to share their insights and experiences. We'll now begin the plenary opening lectures titled Transforming Vaccine Development, National Leadership and Global Innovation in mRNA Technology. To chair this session, I am pleased to invite Professor Kang-ho Lee, Senior Advisor to IVI. Professor Lee brings over 28 years of distinguished service in public policy. He has held senior leadership roles in Korea's Ministry of Health and Welfare, as well as the Ministry of Economy and Finance, including Director General of the Health Industry Policy Bureau and Deputy Minister of the Global Vaccine Hub Office. He played a pivotal role in Korea's successful bid to host the WHO Global Training Hub for Biomanufacturing in 2022, helping to build training capacity for low and middle income countries. In addition to his government work, he is a professor at the KAIST Graduate School of Future Strategy and co-director of the Future Strategy Research Center. He holds degrees from Seoul National University and Harvard Kennedy School. Please join me in warmly welcoming Professor Kang-ho Lee.
Good morning. Distinguished guests, ladies and gentlemen, my name is Kang-ho Lee, Senior Advisor to IVI. It is my great pleasure to chair the opening plenary lecture session, Transforming Vaccine Development, National Leadership and Global Innovation in mRNA Technology. I am honored to introduce our second speaker, Dr. Sanjay Singh, who is joining us virtually from India. Dr. Singh, can you hear and see us clearly?
Yes, I can hear you very clearly.
Oh,
Is I clear?
Yeah, I can hear you. Dr. Sanjay Singh is the CEO of Geneva Biopharmaceuticals Ltd. Notably, he led Genova to develop India's first indigenous mRNA-based vaccine for COVID-19. Today, he will address breakthroughs in vaccine technology, enhancing cellular immune responses with self-amplifying mRNA platforms. Please welcome Dr. Sanjay Singh.
Thank you. So, good morning from India, and thank you very much for inviting to this meeting. And today, I'm going to talk about the breakthrough in vaccine technologies and how it is important from mRNA perspective, because we feel that cellular responses are also very critical, and the technology which we have developed based in self-amplifying mRNA is contributing towards that side. So if we want to say in general, vaccine technology is one of the most important technologies developed by humankind, which has saved hundreds of millions of lives, not only in terms of individual, but also in terms of communities. It has saved communities. And if we start from the discovery of the vaccine or application of the vaccine, we can go back more than a century, and where the first generation vaccine was developed at UK by Professor Edward Jenner, and it was in form of whole cell or whole organism, and that technology continued till today. And even we can say that hundreds of millions of people are getting vaccination in form of polio vaccination all around the world based on the same technology. Rabies is one of the examples, if I want to say. But with science improvement, science going forward, some limitations were always in the present technology, so you have to enhance that. You have to take the next level. And I say second generation technologies was created in form of antigen-specific vaccine. And here recombinant DNA technology added a lot of input into it. And we can see one of the most successful vaccine, if I want to name personally from my choice too, hepatitis B and HPV vaccine, which is again used today in millions and millions of cases. Further, science improved, and we wanted to have more specific delivery system, and it introduced the nucleic acid vaccine technology in close to 25 years ago. And in that, it has added either DNA or adenovirus-based vaccine. And one of the vaccine we can say came during COVID. It was AstraZeneca vaccine for COVID-19. But in last 5 years, and which is the relevant topic for this conference, is mRNA platform appeared, and it appeared because of some need-based innovation which came here. So vaccination is used all through, starting from infancy days of a child until the older age. But this COVID-19 has affected the worst in the people who were old or who were having comorbidity. So more than 81% of the global deaths during COVID-19 was in this population, older population or the population which was getting affected. So mRNA technology at that time, these were two leading organizations. One was, as given in the slide, NIH and Moderna, and then BioNTech and Pfizer. They went through this technology in which use this modified substitutions of bases, and they used non-replicating technology. And the first vaccine which was approved was BioNTech vaccine, and again it went into hundreds of millions of people. Same with the Moderna one. But at that time, we were also into the development phase in India, and we decided to follow a different path, and we decided to follow the path of self-amplifying mRNA. Why the difference between self-amplifying and non-replicating is given on the right side of the figure. Here, there is the presence of antigen for much longer period, more than 3 weeks. In case of replicating, non-replicating, within 18 to 20 hours, the protein is gone. The reason behind it that there were two specific possibilities. One, from immunobiology point of view, if the antigen is present for a longer period of time, your B cell mature to make right set of antibodies. And the second was that you might have to give a very low dose, and which is very important because this is a new technology going to people. So this is the way we decided to follow the path of self-amplifying mRNA. But the vaccine, both which were developed in US, it was having another limitation, and the limitation is they were at a very low temperature. So the whole program for Genova developing self-amplifying mRNA platform from day one was from the term of, can we democratize this technology platform? Next one. This is one of the biggest reasons that only 95% population was not feasible to supply this vaccine because need of very low temperature. And if you see from the left side, what is the mean temperature, and we don't have, including in India, we don't have the whole supply chain system, which is on the ultra low cold temperature. And we have the systems everywhere in the world, 2 to 4. So it is very, very important if you think about democratization of this vaccine platform technology. The first problem to solve, can we make it stable at 2 to 8 degrees centigrade? Next one. And the second was ease of manufacturing. Still, we believe that, yes, COVID has come. We have made a lot of effort. Great organization has made a lot of effort. But the reality is that we have not reached to the most of the corners of the world. If a pandemic comes, how we can do the manufacturing. So for ease of manufacturing, another technology we conceived and developed and moved it forward, that it doesn't require high-speed homogenization. It is having two independent components, cLNEs and mRNA, mixed together and go ahead on it. So this is easy to produce in developing countries, and we can create different kind of regional centers where we can go ahead. And also that we backward integrated because we face a lot of difficulties in getting raw materials. So more than 90% of the raw material today we produce back in India itself. Next one. The third technology which we decided to adopt into our vaccine technology was the intradermal delivery system. And most of the audience must be from the immunology point of view. Our whole defense system is below the dermis. It is not intramuscular because the delivery system was not available. So we are using, and still we are using the intramuscular delivery or some different bit. But the real defense system for the body is below the dermis. So we decided to use intradermal vaccine delivery. And I was part of some research many years back where WHO and PAS decided to, and we found that intradermal delivery system is much efficient, but technology was not available. So we brought this technology also into the game. Next one. So here we, if you talk about the culmination technology, we brought 4 technologies together one by one. It started from self-amplifying mRNA technology, moved to the wrapping of the mRNA through nanolipid. The third, we brought the lyophilization because stability of mRNA is only affected because presence of water. Water go and hits the phosphodiester bond and it breaks the bases. So we lyophilize, remove the water. So it is stable even at room temperature for 6 months. And then we brought the technology of intradermal. So basically, 4 technologies we mixed together and finally created the vaccine. And again, we say that this was a very serious effort to us taking this platform to democratization. Next one. So 2 vaccines we developed. The first base vaccine was basically intramuscular. We went into it. But the second vaccine was using this specific device, which is jet device, needle-free device. And when we have immunized thousands of people, this is much faster and efficient to deliver as compared to the intramuscular. And this work was published in Nature Medicine last year. And the second thing was we further gone ahead because of one of the limitations for this vaccine, both the vaccines which was approved, non-replicating, were that their immune response was going down very fast and cellular response was limited. Next one. So here, if we say that today vaccines, so the 3 types of these vaccines are there, but I don't want to go into all that. I just want to focus on the doses. So it is 0.5 ml 100 microgram was Moderna. At that time, 30 microgram for Pfizer and 0.1 ml 100 microliter 10 microgram of our vaccine, and which was based on different technologies. But we were not having limitation of addition of any PEG because PEGs induce some reactogenicity. So we don't have the PEG, and our storage temperature is 2 to 8 degrees centigrade. So this is, if we say in nutshell, what is the difference between 3 technology in terms of the vaccine specification, this was the difference. Next one. Now, this is very, very important slide in terms of data. We wanted to see that what kind of functional response in terms of T-cell responses are there. So we went ahead and we took the PBMCs 90 days after immunization, and we tested different formats, which is interferon, TNF, PAP, in terms of alone and in terms of combination. And you can see as compared to ChAdOx, which is the AstraZeneca vaccine, it's a very strong vaccine, but our polyfunctional responses were alone and in combination were much superior. And this was one of the biggest advantage which we found. And if we see on the right side of the box C, you can quantitatively say that the significant response came in terms of mRNA vaccine, which is known as very soft vaccine, but cellular response was very, very good in terms of when we went for 90 days after vaccination. Next slide. This is very, very important slide. Again, these are 2 data which is very close to my heart because how long memory response you have. And for that, we tested activated immune biomarkers. If you see in the side of A or F, you can see a statistical big response in case of CD4 memory T cells and also CD8 memory T cells. both this. And if you further want to explore, you can see on the E parameter, there is a huge difference in terms of statistical difference in terms of long-term memory. And these two slides shows in combination that we have cellular immune response in form of T cell responses, but we also have long-term memory responses. 9-month data we have done. Similar results are also coming into that. And this is taking added value into it that self-conforming mRNA immune response is quite different than non-replicating vaccine, mRNA-based vaccine response, especially in terms of cellular response, because when new exposure comes, you can have immune response available to you. Next slide. Our collaborator, I think he's one of the speakers today, Professor Dan Baruch. He wanted to test our vaccine head-to-head. It is not in terms of very unique, but in terms of scientific, this was the first opportunity when a third party had tested our vaccine head-to-head in terms of cellular and humoral response, both. And here the 5 doses was tested from 0.03 to 3 microgram. In mouse setting, he will explain much better when his time will come. But we can see a statistically significant response and even 10 times lower dose. So what we started with envisaging that how the responses will come. So here dose is very small to give even 10 microgram, and which is giving much higher response, especially if from these slides, either binding antibodies and neutralizing antibodies or CD4, CD8 responses, you can say the potency of this vaccine is around 10x. And more importantly, he carried half across the world this vaccine into his backpack, and then he tested it. So this was very soothing to us that this kind of data is there, because none of the mRNA vaccine was approved in India. It was not available, so you cannot do the testing on this. And it has been done into his lab. And during COVID, I think he was most popularly published during the COVID-19. So they have done it. Next slide. So if total together we put it, there is an intradermal vaccine, which enhance on the left side, the watch slide, if you send, which enhance your immune responses, and which give the polyfunctional immune response. And it is very important, not only humoral, but polyfunctional cellular responses in terms of infectious diseases, in terms of oncology, and in terms of other possibilities which we are exploring at present. And this came into one of the research commentaries from Nature, and we said it paradigm shift in vaccinology, harnessing the cellular immunity. Next one. Now, if we talk about the platforms, I think this is what is the topic for this meeting was. So we developed 4 independent platforms. The first platform was plasmid. So this, we, as a biopharmaceutical company, we have making few products for the commercial purpose. So we expertise this plasmid DNA platform, and we can produce in kgs so this is one where we focus our research, and we fix that from research grade to CGMP grade can be produced ample quantity of plasmid. Second, we focus on the lipid nanoparticle, and without any patent infringement or legal cases, that today we have the very open and clean, because this mRNA technology is full of the whole patent landscape. So that is another platform we developed, and these are independent manufacturing processes and components. The third platform is mRNA platform, which is well known, but only thing we are self-amplifying mRNA. And fourth is the testing of these vaccines is very important, that how this individual research program or overall vaccine potency you are testing, it is testing in the mammalian platform assay system. So that is because when you are giving the vaccine, it is interacting with human or mammalian platform system, and there are assays we develop. So these are the 4 independent platforms we developed for the mRNA vaccine technology. Next one. And if we put together today, so how this technology looks like, we put kind of funnel where everything else is common, which is enzymes are common, cofactors are common, nucleosides are common, lipids are common. Only the change in the antigen-specific sequence you can put, and you put together, and you make the vaccine. Why it is so important for future and democratization point of view, because we believe there should be regional center where these materials can be stored. You can make mRNA, you can make CLNE, and at the time of need, you can ship it to different regional centers. What WHO was very seriously thinking about mRNA hub. We used to say the term regional hubs. And you can mix together, lyophilize together, because this technology existing in most of the part of the world, and you can finally formulate the vaccine very quickly. Because practical experience I can see from developing the vaccine, making the vaccine, passing through regulatory rules, shipping to different countries, it's a quite cumbersome process. And I think as a community, we have to focus on it. If we have to democratize this mRNA platform technology, what are the different components, how the manufacturing capabilities we can enhance across the globe. Next one. So today, we are trying this technology for different platforms with the support from CEPI. We are using this platform for rabies vaccine. Another critical factor I want to mention that till the virus is there, I think disease will continue. One of the examples is in Australia. Australia banned bringing any FMD virus into the country 30 years ago. But in the last 2 years, they had around 13 or 14 outbreaks. From where this virus is coming? Because it is live attenuated, and if any small quantity of it is not completely attenuated, there's a possibility of outbreak from the vaccine itself. So hopefully with CEPI's support, we may reach the global aim of eradicating rabies by 2030. We are making very well, and I'm very thankful to CEPI to giving this support to make the rabies vaccine. The same way we have now partnered with CEPI based on the progress which they have seen on the rabies Nipah vaccine. And this is also supported by CEPI. The third vaccine, I think those two are easy one. The third one, which we have gone into the most difficult area, and this is difficult, nothing has achieved in the last 100 years. But we have said we can use this platform for TB vaccine, and this is in collaboration with Professor Dan Baruch's lab at Harvard. We are very happy with the progress we have made, and we are moving soon into the clinical trials. And the last one is Mpox, because country needed it. So we are doing it in collaboration with Indian Council of Medical Research. And again, here, some of the research which has added fuel to us from Dan Baruch lab, that there are 2 antigens are required, not all the 4 antigens are required. So these are the 4 vaccines which we can say that program we are working. And again, very thankful to CEPI, to Harvard Medical School, and ICMR for support. So I think this is my last slide. Thank you very much.
Thank you so much, Dr. Singh, for your informative, inspiring talk. I believe the Geneva played a significant role during COVID-19 pandemic, leading the way in mRNA vaccine development. I wish for its continued growth. Now,
thank you.
Dr. Singh would be happy to take questions from the audience. Please raise questions to Dr. Singh. Okay, gentlemen.
Congratulations. I'm Junri from Jeonnam National University, Korea. As far as I know, many leading mRNA vaccine companies tried self-amplifying format at earlier stage, but they all ended up with linear mRNA. vaccine. You should have your own, know-hows or breakthroughs. Can you tell us what your, breakthrough was with, self-amplifying platform?
Thank you. Very, very genuine question. So by profession, I'm a scientist. I was at NIH, and I was next door neighbor to Barney Graham. And I left that and came and started this company. So there are two very independent components. One is research, which runs very well in 50 microliter or 2 microliter ep drop. But when you convert into 1,000 liter, this is the different life. So for last 15 years, we have taken 7 products, which is biopharmaceutical product to market. So this scale up is a very, very unique thing, and which is not available in the academic setup. So industrial setup is a total different setup, total different thought process. And to support my view, I predicted that Pfizer vaccine will go first to the market, though the NIH and Moderna both were there, and it made it to the market because manufacturing muscle is a different kind of art. So this is one of the difference we want to say. And the second of the difference, somehow these are complex problems, so you have to comprehend it all together. And what is meaning comprehended? From the sequence to how it will be shipped to the market and how the phase 4 data will come. If we don't have this whole picture, then it is difficult for individual company or institution to do it. So I think we were at a unique position, and it is not that COVID-19 brought us to mRNA field. We were looking into immuno-oncology from 2 to 3 years before, and where we believe that cellular responses are very, very important. So we were in the field, but COVID-19 moved us a bit faster, and this is the platform has come. I think I have tried to answer your question.
So you succeeded in a scale up and solve the manufacturing, large scale manufacturing problems with self-amplifying platform. Is it right?
Yes, we can produce 1 billion doses.
Thank you.
Okay, next question. 네, 한혜 김.
Hello, Sanjay. This is Hanne Kim at the Right Foundation. Thank you for your presentation. I found your data comparing the level of neutralizing antibody titers and the degree of antibody binding very impressive, especially compared against what you see from the competitor vaccine from Pfizer. Could you help us better understand potential mechanism by which your immunogen can achieve that? Is that related to something inherent about your self-amplifying mRNA? Or is it a combination of that and how that's presented with your lipid nanoemulsion?
Yeah, thank you very much. So, Honey, we were knowing from day one that non-replicating antigen is only available for 16 to 18 hours maximum, and it will go away. And once you give in 16 to 18 hours, the availability for processing of antigen or availability of enhancing the immune response is very, very limited. So it'll get a very peak quick and it will go away. But in our self-amplifying mRNA, antigen is available roughly between 21 to 28 days, what data we have found. And availability of antigen makes two different things. It keep on boosting and reminding it, but it keep on maturing the immune response. So this is the hardcore if we go back into immunology. So this is why the two very successful vaccines in case of hepatitis B, which is nanolipid particle, which is consistent again for close to a month, you can see the antigen. And for HPV, both vaccines were very successful. But so the knowledge which we brought into this, it is from the previous available functional immune response and vaccine technology we built together. And this is what we believe is making the real difference. This is my answer to it.
Okay, next question. Okay, old lady.
Yeah, thank you for your nice lecture. So I'm Dokeun Kim working for IVI. So I have a question about your lipid. So you said your mRNA is just absorbed to the lipid, not encapsulated. So I'm wondering then, the conventional mRNA also is absorbed to your lipid, and the efficiency is increased or the same? Also, why do you use this absorption not to encapsulate it?
I will give into this answer in three parts. So the first part is when we mix the nanolipid in components and mRNA and throw it for capsulization through high pressure, around 30% mRNA get degraded, so there is a lot of loss. So here in our case, our yields are more than 95% because we just mix together by positive negative charge. If our mRNA size would have been 4 kb, then stability could have been problem. But our mRNA size is around 12 kb, so it gets very much encircled outside the lipid. So this is the component we discovered, we made it, and the biggest time gone to us into the whole developing this vaccine platform, how to mix these together. So I think some genetic information available for physics. The physics came and solved this problem, how to mix it. And what is the ratio to mix it? What is the quantity to mix it? NP ratio, how to do it. So that kind of science we did, and that stabilized this in this form. This is part one. The part two, if we have to make this vaccine by microfluidization, this technology is a little bit difficult. It needs some additional steps. And if we want to make somewhere in Kenya this fill and finish facility, we have to just ship the lipid separate, nanolipid separate, which has 2 years stability. We can ship mRNA separate, which has 2 years stability, and just mix together and lyophilize it, so it go on. So this was very deliberate attempt to do this kind of research and bring this kind of technology. Again, from the question of can we democratize this technology? Because we believe our vaccines are first generation vaccines. I can say not non-replicating first generation, we can say 1.5 generation, second generation. But still there is a lot of hope and scope to take it forward to the real development into the mRNA platform technology, and which at present we are doing with CP support. We are working with Dan, and we are also going to have some opportunity to work with few other major organizations around the world to take this technology forward. So still we have done it, but still there is a huge scope to take to the next level.
Okay, thank you. Next question. There is no more questions. Thank you, Dr. Singh, for your wonderful talk and the insightful answers. Please give him a big round of applause.
Thank you. And my special message to Jerome that sorry, because of my pre-commitment, I was not able to come, but very soon I will come and see him in person. Thank you very much, and thank you very much for this invitation.
Thank you. This concludes the opening plenary lectures. Thank you very much.
Thank you very much, Professor Lee. We'd now like to invite all of our speakers and dignitaries to the stage for a group photo. So if you are sitting behind a name tent or a reserved name tent, we'd like to welcome you to please join the photo. While we are clearing the stage, I'll just make a quick announcement that to access the Wi-Fi in this room, the name of the network is called Four Seasons, not Four Seasons Meeting. So there should be no password for the network Four Seasons. And may I now ask our speakers and distinguished guests to please join us on stage for the photo. Thank you. We'll now transition to session 1 of the forum, Accelerating Vaccine Innovation Through mRNA and Novel Platforms. To chair this session, I am pleased to invite Dr. Mangi Song, Deputy Director General of the Science Unit at IVI. Dr. Song is a leading expert in virology and vaccine development with a deep commitment to addressing public health threats in low and middle income countries. He also serves as an adjunct professor at Yonsei University College of Medicine. Early in his career, Dr. Song focused on viral diseases such as HIV, HCV, and HBV. Since joining IVI, he has expanded his work to include vaccine development for norovirus, hepatitis A, SFTS, and MERS-CoV. He led an important effort in partnership with CEPI and the UK's NIBSC to establish international standards and assays for MERS-CoV. During the COVID-19 pandemic, Dr. Song played a critical role in developing qualified ELISA and FRNT assays for clinical analysis, contributing significantly to the development of SK Biosciences' Sky Covione vaccine. Please join me in warmly welcoming Dr. Mangi Song.
Thank you very much. Good morning, everyone, and thank you for joining this session 1. Accelerating vaccine innovation through mRNA and novel platforms. We are very pleased to have you here today, and we are especially honored to welcome distinguished speakers who will share their expertise in vaccine development. Let me briefly introduce today's first speaker for session 1, Dr. Martina Oakes. Dr. Oakes is a highly respected vaccine researcher and currently serves as a project lead at CEPI, where she oversees a key program in mRNA vaccine development. Her previous work at Sanofi Pasteur involved leading international research collaboration on pneumococcal and pertussis vaccines. With her strong expertise in immunology and global health, she continued to play a central role in advancing next-generation vaccine platforms. Please give a warm welcome to Dr. Oakes.
Yeah, thank you very much to the organizers. It's such a pleasure to be here today. And I will present CEPI's strategy on mRNA and new vaccine technologies. First, just a little reminder of how this was with COVID-19. So the graph demonstrates really the difference it can make when you can have a vaccine ready. After 326 days, the first vaccines were licensed, and you can see the infectious curve prior to these 326 days, and what is marked in with the dotted line in blue is 100 days. So you can imagine if you would have had a vaccine at 100 days rather than at 326 days, how different the pandemic would have progressed. And that led CEPI to develop the 100-day mission, in which if a vaccine is ready at 100 days for initial authorization and manufacturing at scale, then it can be used when appropriate. And that requires really a shift to preparedness rather than just response and rollout of vaccines. So we really need to invest in front-loading and breaking the firewall between development and intervention. In that, rapid response platforms play an absolutely crucial role. So what are these rapid response platforms? They're technologies that have the same basic components as a backbone, where you then just insert new genetic or protein sequences, and they can then be rapidly used against disease X. So the concept is really the plug-and-play concept. We at CEPI as well developed a disease X product characteristic profile, and it's evolving. This is a target. It's not really in the same way a fixed target as you may have for an established disease. This is where we want to be in our preparedness and in the technologies we use for the 100-day mission to be ready for a disease X. So the preferred is that you have cold storage at refrigerator temperature, that you have a shelf life of 1 year, that your technology allows single-dose vaccination, ideally needle-less intradermal administration or oral administration, intranasal or other devices, hopefully with a very high efficacy and hopefully applicable to target populations 1 year and upwards, including immunocompromised or pregnant women or healthcare workers, and ideally a 1 year durability of immunity. So this is really the ideal. I think very few platforms are there today that actually can make that, meet that ideal, but this is what we are ultimately would like to see. How do we use platform? Just a couple of definitions so that we're all on the same vocabulary. At CEPI, we use the term modality for the form of the structure of the active substance. So is it mRNA, is it protein, is it viral vector? Platform is the final product of a company. So it is the drug substance and the drug product together, and platform technologies are the technologies that lead us to make a drug substance or a drug product. So one platform could have multiple platform technologies, but one modality. In the 100 for the 100 days mission, we invest in protein subunit platform, we invest in RNA, and we invest in viral vectors. We do not invest for the 100 days mission into the other vaccine modalities that you could imagine or that are used for other diseases. We as well look now at platforms holistic. So we have developed these dashboards where we interrogate potential platforms for their adaptability. So is it a plug-and-play concept? What is the process consistency and so on? Where we look at compatibility with different technologies, viral family, population, and so on, where we look at suitability in terms for the disease X. Is it thermostable, cost of goods, durability of protection, those type of parameters, where we look at the regulatory status of any given platform. So is prior knowledge available? Has an authorization been achieved? Where we look at the manufacturing for a platform, so at capability, capacity, scalability, supply chain, but as well, then we look at the facility, GMP compliance, analytical capability. So we are not just looking at one aspect, we are not looking really holistic what any of the developers with who we are working with can actually provide. This is just how we score this then. We just make green, yellow, red scoring. It's not detailed enough to really understand the platform, but it gives us a holistic overview. And this is just an example, and you can see, for example, that the middle platform, the one in 2, is actually in many regards in pretty good state of readiness. There are some gaps to fill that are still yellow, but there's as well highlighted in red, for example, that the intellectual property is not quite where it should be, that specific developer wouldn't have the freedom to operate. And that, of course, is a hindrance when you have to go to market. Just a reminder of COVID-19 pandemic triggered vaccines that actually advanced quite far. So advanced platforms where Moderna, BioNTech, Arcturus, and Daiichi Sankyo as well have now received authorizations by stringent regulatory authorities. Emergency use authorization was given to 4 developers: Genova, you heard Sanjay talking earlier about this, Valvax, DemiRNA, and CSP Pharma. But there's as well still in phase 3 development, so in really advanced clinical development is VLP Therapeutics. And that was the status of June 2024. I don't want to guarantee that, that this is the status of today, but this is how it was a year ago. Just to recap on what is actually in the pipeline, you can see this is the CEPI internal analysis that we did last year. And again, it may not be entirely complete, and that's not the objective. The objective is to give you a little bit of flavor of where we are. So this is divided by modality and delivery vehicle, and you can see that the preclinical space is quite advanced. There's a lot of technologies in the preclinical space. There's some in the phase 1, phase 2 space, and there's some very few in the later stages. And you can see that in terms of the RNA modality, so in red is circular RNA, in the one beneath in the blue, dark blue, is modified linear RNA. The one in brown is self-amplifying RNA. The one in green is non-modified linear RNA, and then there are there's few trans-amplifying RNAs, and there's quite a number where we were unable to find out what the exact RNA modality is. So it's quite a lot of different technologies that are really being developed. Quite similar for the delivery vehicles. The vast majority is still lipid-nanoparticles, but there is an increasing number as well of non-lipid nanoparticles, other technologies, and as well, again, a large number of technologies where we were unable to identify what the exact delivery modus is. These are the different areas where CEPI is investing. We are looking at RNA modalities and delivery innovations, where we currently have 11 projects signed. We are looking as well at enabling technologies. So they are technologies that are more platform agnostic, that are like algorithms to optimize the DNA template or synthetic DNA, this type of technologies. And we currently have 2 projects signed under what we call the enabling technologies. Platform development, that is more the first generation linear mRNA with lipid nanoparticles, and we are just, many of the projects are just using the technologies for CEPI's priority pathogens, like Lassa, Nipah, Rift Valley fever. And there we have 9 projects currently signed. We are investing quite a bit in manufacturing innovations as well. We have 10 projects signed, and then we have as well a manufacturing facilities network where we currently have 4 partners signed. In terms of the different modalities as well, the CEPI is investing in quite a number of different modalities. We have with conventional mRNA technology, so like BioNTech, Pfizer technology, we currently have 13 projects. We have 4 projects that utilize self-amplifying RNA, We have one project that utilizes trans-amplifying RNA and 2 that utilize circular RNA. And the majority of those, as you can imagine, is in the preclinical space. In terms of delivery, we are looking at different delivery methods as well. We have 11 projects in the traditional LNP space. We have 6 projects that are non-LNP delivery. And then we have as well 3 projects that we call the next generation LNP. These are the LNPs that are tissue targeting or polyethylene glycol free, that type of advanced LNP technology. Now I'm switching a little bit to more details on the projects we are investing in. So we have with Wellcome Leap as well a partnership that we started in, the project started in 2022 and was called R3. The project was 3 years. It has evolved since then to become what is now called R3 Global. R3 stands for RNA Readiness and Response, and is a project that is looking with multiple partners at continuous production of RNA, as well as the treatment against different conditions ranging from cancer all the way through to Zika, including bacterial vaccines as well. It's now focused on building a network of biofoundries, basically CDMOs for formulated RNA, and wants to do that in an economically sustainable on-demand capacity. So this is an ongoing project with Wellcome Leap. In the manufacturing innovations, we are looking at different areas as well. As you may know, CEPI works by calls for proposals. Our investments are we publish a call for proposal on our website, we receive applications, we select applications that fit to our strategy and that match otherwise what we are looking for in our developers, and then we award according to those applications. And we had calls for proposals that were addressing speed, so that are really 100-day focused. Then we had call that focused on scale, easier scale up or scale out, flexible, continuous manufacturing, AI facilitated systems, that kind of thing, and that focuses a lot on what actually you do after you met your 100-day timeline. So really when you really enter manufacturing and roll out. And then we had one that was more focused on access, which is what is necessary for the technology to actually meet what you need to do to go in LMICs. So you need to have thermostability, you want to have reduced cost of goods, you want to have ease of administration. In terms of the manufacturing technologies, here just highlights where we are investing. We have signed with the University of Sheffield. This was originally coming from the R3 program. It did not go into R3 Global, so it was not continued in the collaboration with Wellcome Leap, but we signed it as a CEPI project. We have a project with Eubiologics, which is on AI to identify optimal operations of process modules to manufacture RNA and LNPs. We have a project under this R3 Global program together with Wellcome Leap with Centillion, which is modular, integrated, multipurpose, and automated RNA microfacture. This is basically continuous manufacturing of RNA LNP. Similar with RiboPro, we have a project there as well under R3 Global. This is continuous manufacturing, and it contains as well a DNA amplification step. And then we are working now with Ethris on establishing automated RNA manufacturing processes. And I think that we have so many is as well just a reflection of really how much is going on in this space in terms of development of different manufacturing technologies for RNA and formulated RNA. I mentioned the enabling technology. We are investing in synthetic DNA. We have 2 projects signed, one with DNA Script, which is really a completely synthetic DNA manufacture, and they are currently not GMP ready, but they are able to produce linear mRNA suitable for the manufacture of RNA. And we are working as well with Afrigen, who is collaborating with Syngoy on the development of a synthetic DNA process. I mentioned the manufacturing network. This is just highlighting the facilities we are working with. What is highlighted in red are facilities that build RNA technology capabilities. It's basically Biomaningos in Brazil, BioNTech in Rwanda, the Serum Institute of India, and Biopharma in Indonesia. And now I'm just coming to the calls for proposals that we currently have open in case you are interested in applying. They are published on our website. So we have one that is really a very, very broad call for a broad range of vaccine innovations for pathogens with epidemic or pandemic potential. And there are 2 focus areas open. One is for innovative vaccine platforms, and the other one is for vaccine candidates. And there, the vaccine candidates is now limited. We only accept applications for Lassa vaccine candidates or disease X candidates. In the past, we had just open to some other diseases, but that is now closed. And just last, I would like to highlight a call for proposal that is currently as well open on analytical technologies for vaccine release speed, cost, and development. And there we are looking for applications. It's not limited to RNA technology. It's as well for other vaccine modalities to reduce the time to vaccine development, to reduce the bottlenecks in the analytics, basically. And I think that was my last slide, so I thank the audience for your attention. Thank you very much. We are doing questions later.
We'll take a question after the Denver's presentation together. Thank you very much, Martina. Now I'll introduce the second speaker for this session. It is my pleasure to introduce Dr. Denborough, Professor of Medicine at Harvard Medical School and Director of the Center for Virus and Vaccine Research. Dr. Borrow is internationally recognized for his pioneering research on vaccine development against major viral diseases, including HIV, Zika, and COVID-19. He led the development of the Johnson & Johnson COVID-19 vaccine, and his work on HIV vaccine candidate has shaped the global scientific agenda in this field. Please join me in welcoming Dr. Denborough, please.
Thank you very much. And it's really a pleasure for me to be here today. Thanks to IVI and the conference organizers for the invitation. And what I'd like to do in the next 20-25 minutes is to talk about correlates of protection for respiratory virus vaccines with a focus on some recently published work with COVID-19 and some unpublished work with influenza. Here are my disclosures. So it goes without saying that the development of the COVID vaccines globally was a stunning scientific success, with an estimated over 20 million lives saved by COVID-19 vaccines globally in the year 2021 that really focused on multiple different technologies, including viral vector platforms and mRNA platforms. The mRNA vaccines, which is, I think, the main subject of today's conference, have shown incredible potential. They have a high degree of protection against severe disease, as shown on the right. But there's several limitations as well. And these are data from the North Carolina Department of Public Health and about 10 million individuals in the United States and North Carolina. While there's a high level of protection against severe disease and hospitalization, it does tend to wane relatively quickly over time, so there's a durability limitation, but also the protection against acquisition of infection is far lower than protection against severe disease. So in this, with the bivalent vaccines in the 2022 to 2023 year, it was estimated to be less than 30% at peak and going down to 0% after about 4 months. So we were asking the question, from a scientific perspective, why would a vaccine give high protection against severe disease but relatively limited protection against upper respiratory infection, which is essentially acquisition of infection? And so we had a hypothesis that intramuscular vaccination typically does not induce mucosal immunity or does not induce sufficient titers of mucosal immunity for protection. And while protection against severe disease and death is probably the most important goal of vaccination, then the public really wants a vaccine that prevents you from getting sick and prevents you from getting infected. So we think the development of respiratory virus vaccines that actually can block infection is actually important to reduce vaccine hesitancy and to provide better efficacy. So we published a paper last fall in which we demonstrated that mucosal boosting can enhance vaccine protection against SARS-CoV-2 in the macaque model. And so in this study, we used a cohort of rhesus macaques that we had, that was started as a collaboration with J&J with the development of their Ad26-based vaccine that received the Ad26-based COVID vaccine 2 years prior to this study and received a boost a year prior to the study. And so this study really began as a boosting cohort in animals that were previously primed. In this cohort, these animals were then differentially boosted. The first group received the Ad26 vaccine by the intramuscular route. The second received the Ad26 vaccine by the intranasal route. The third group received the Ad26 booster by the intratracheal route. And we do think that this is a clinically translatable modality that could be done in humans either with a more aggressive nasal spray or possibly by aerosolization of vaccines, which is being done in clinical trials now. The fourth group received an mRNA vaccine from Pfizer given intranasally. And I'll just say that the current generation of mRNA vaccines really don't work given mucosally because they lack the biophysical stability to do so. The fifth group received no boost, and the last group was a sham control group for a challenge that was done 4 months later. So here are the mucosal antibody responses in the groups that received the Ad26 vaccine given intramuscularly, intranasally, or intratracheally. What I'm not showing in this slide is the serum antibody responses, which were extraordinarily high with all boosted groups. That's in the published paper. I didn't have time to show all the slides. So while the serum antibody responses were strikingly high for all the groups, then the mucosal antibody responses shown in this slide were not. The intramuscular group raised only minimal mucosal responses in bronchoalveolar lavage on the top and nasal swabs on the bottom. The intranasal administration of the vaccine was only slightly better, but if we give the vaccine intratracheally, then we see a stunning increase in mucosal immunity. That's high titer, relatively consistent, as well as durable for at least the duration of the study, which was 12 weeks before challenge. This was seen in BAL, less so in nasal swabs, but also a clear boost in nasal swabs. If we look at mucosal IgA, then we see even a more clear difference. This is really just a black and white difference, that if we give the Ad26 vaccine intramuscularly, we see only a very small blip of mucosal IgA in BAL and nasal swabs. It's only slightly better given intranasally, but if we give the vaccine intratracheally, we see a consistent high titer IgA response in BAL and to some extent in nasal swabs. If we look at T-cell responses in the lung, CD8 T-cell responses in the bronchoalveolar lavage on the top, and CD4 responses in the bronchoalveolar lavage shown on the bottom, the vaccine given intramuscularly gives low-level transient responses for CD8 and really no detectable responses for CD4. Given intranasally is only slightly better, but if we give the vaccine intratracheally, we see high titers of both CD8 and CD4 responses in the lung that are sustained over time. So following challenge, then these mucosal immunity differences really played out in a very striking way following challenge. So if we just go sort of counterclockwise, the sham controls show the expected viral profiles in macaques following this Omicron challenge with about 5 logs of virus that's controlled in about 10 days. In the group that did not receive a boost for over a year, there was still some degree of protection, likely due to long-term T-cell responses as well as some antibody responses. And so in the no boost group, the viral loads were still reduced by about a log. The mRNA intranasally did not work immunologically because the current mRNA LNP formulations are too unstable to be given by the mucosal route, and I think that is a challenge that I think some of the speakers already this morning are trying to address, such as what Martina and Sanjay were discussing this morning, potentially making a more stable version of mRNA might solve this problem. But the current mRNA versions really cannot just be given intranasally. The Ad26 vaccine intramuscularly gave a slight improvement in protection. Intranasally gave better protection, but the Ad26 vaccine given intratracheally was really just a black and white difference. There was really no virus detected except in a single animal for one time point. So this is, we call this near complete protection against acquisition of infection. In nasal swabs, we saw a very similar result. In the sham controls, there were 5 logs of virus that was controlled over 14 days. In the group that was not boosted, this was reduced slightly. The mRNA intranasally didn't really do any better. The Ad26 intramuscularly also didn't really do too much better. This was slightly improved with the vaccine given intranasally, but the Ad26 vaccine given intratracheally gave a substantial improvement of protection such that virus was only detectable on day 1 following challenge and was undetectable by day 2 following challenge in all but 1 animal. And actually, the day 1 viral loads, we're not even sure that's truly replicating virus. It could just be the input challenge virus itself. So it does appear that if we can boost intratracheally directly in the lung, then we can achieve a level of protection that's really not achievable by other methods. And that appears to be through the induction of mucosal immunity. The multivariate statistical analysis also showed that the mucosal antibody and T-cell responses are really the top correlates of protection and not the serum systemic antibody responses. So in another study that we published last fall, we looked at the correlate of this in humans. And in humans, and this is a clinical study in humans now, an observational study, in which we looked in Boston at a cohort of people who received the Moderna and the Pfizer mRNA vaccines. And here we see, like dozens of other groups have shown, that this was in the 2023-2024 era with the XBB.1.5 vaccine, that this vaccine given intramuscularly in humans results in a substantial increase in neutralizing antibody titers to XBB and all of the variants through JN1 by more than a log. So a very nice increase in serum neutralizing antibody responses. Also, this vaccine also only minimally boosted the ancestral strain, showing that this monovalent vaccine appeared to overcome some of the immune imprinting issues that were present with the bivalent vaccine the year prior. So we think the monovalent version is actually a more immunologically robust formulation than a bivalent version that includes an ancestral strain that no longer exists. So the serum antibody responses we thought looked really quite good. However, there were no mucosal antibody responses in nasal swabs in humans following mRNA boosting. And so this is really an exact mirror image of what we saw in macaques, that the intramuscular boosting with mRNA vaccines gives substantial serum antibody titers, but really minimal to no detectable mucosal antibody titers seen in humans. And we know that the assays are working because in the small subset of individuals that had an intercurrent SARS-CoV-2 acute infection, then there were high neutralizing antibody titers in serum nasal swabs. Not shown here, but in the paper, there is some modest level of mucosal IgG, but really minimal mucosal IgA, and no detectable neutralizing titers. So the data from the COVID-19 vaccines The data from macaques and humans suggests that both antibody and T cells, and not antibodies alone, are important for vaccine protection. The field is really focused on short-term neutralizing antibody responses, and while these are important, we believe that the durability of neutralizing antibody and T cell responses are really the critical parameters for protection against severe disease with viral variants. I didn't have time to really go over the third point, but I mentioned it briefly, that we think the bivalent boosters in the 2022 to 2023 era were limited by immune imprinting to the ancestral strain, and the monovalent XBB.1.5 and KP2 boosters in the subsequent 2 years largely overcome this problem. But what I did show in detail was that the current intramuscular vaccines really do not boost mucosal immunity. This is true in both macaques and humans. And mucosal boosting appears to be required for that quantum leapfrog jump of protection against viral replication to near complete protection against acquisition of infection in macaques. So in the second half of my talk, I'll switch to some unpublished data for both seasonal flu and pandemic flu. So seasonal flu remains a substantial problem with a large number of deaths worldwide. In the United States, the two platforms that are approved for vaccines is the inactivated virus vaccine, which at the time of this study was the quadrivalent inactivated vaccine, QIV, now is the trivalent inactivated vaccine, as well as the live attenuated vaccine, the Flumis vaccine, given intranasally. So this is a study that will come out soon. It's a comparative influenza vaccine study in macaques, and the goal of this study was to compare the immunogenicity and the protective efficacy of systemic versus mucosal boosting strategies for influenza vaccines. In this study, we used 42 cynomolgus macaques, and all the groups were primed at week 0 with the QIV inactivated virus vaccine. This is sort of the poor person's approach to generating some degree of influenza immunity in the population of macaques. Now, this is clearly not representative of human immunity that develops over a lifetime with multiple different strains, but it's probably better than nothing. And then at week 8, then we did essentially a single-dose vaccine that was a differential boost. This involved the Rhesusat 52 vaccine, which is a vaccine that we made in-house, given intramuscularly, intranasally, or intratracheally, trying to build on our COVID experience. We also used an mRNA vaccine that was provided to us by a major pharmaceutical company and is the phase 3 vaccine candidate that's in humans. And this is compared head-to-head with the two clinical comparators, which is the QIV vaccine, given the inactivated vaccine given intramuscularly, and the Flumis vaccine given intranasally. And then group 7 was a sham control. And then 12 weeks after the boost was a challenge with 10 to the 6 TCID50 of the H1N1 Wisconsin strain. I should mention that the RAD52 and the mRNA vaccines express the homologous H1 Wisconsin antigen. So this is a seasonal flu with H1N1. I'll also mention that H1N1 in macaques is a non-lethal disease, and so viral loads rather than clinical symptoms was the readout. So these are serum neutralizing antibodies, and please concentrate on the boost because all the animals, all the groups received a common prime, which was the QIV vaccine. So the differences seen between groups is really the result of the boost. So if we go in counterclockwise orientation, the Flumis intranasally really did not result in much of a boost. The QIV boost intramuscularly resulted in about a log boost of neutralizing antibody titers in serum. The mRNA given intramuscularly and the RAD52 given intramuscularly were substantially better than the two clinically licensed vaccines, resulting in about a 2-log increase in serum neutralizing antibody titers. Interestingly, if we give the RAD52 intranasally or intratracheally, then we also get a substantial boost of serum neutralizing antibodies. So mucosal administration of the RAD52 vaccine appeared to increase serum neutralizing antibodies almost as well as an intramuscular dose of that vaccine. If we look at mucosal antibodies, and here we look at BAL IgA responses, then we see something completely different. And it's really a black and white response similar to what we saw for the COVID vaccine, which is that if we look at IgA and mucosa, there really is a flat 0 response with the current clinically licensed vaccines. The QIV and Flumis vaccines do not generate mucosal IgA. The mRNA and the RAD52 intramuscular vaccines generated minimal mucosal IgA. It's slightly better if the RAD52 vaccine is given intranasally, but we see high levels, relatively consistent and relatively durable mucosal IgA if the RAD52 vaccine is given intratracheally. So we see this sort of black and white difference if we can get the vaccine priming in the lung compared with in the muscle. If we look at CD8 T cell responses in the lung, then we also see this black and white difference. The clinically licensed vaccines, the inactivated flu vaccine and the flumis vaccine, really do not generate CD8 T cell responses in the lung. The mRNA vaccine, although it did generate CD8 T cell responses in peripheral blood, only generated CD8 T cell responses in the lung in a sporadic way in 2 out of 6 animals. The RAD52 vaccine did generate CD8 T cell responses in the lung given intramuscularly. They were low and transient and largely disappeared by the day of challenge. These were slightly higher if this vaccine was given intranasally, but if the RAD52 vaccine is given intratracheally, then we see a high level of consistent CD8 T cell responses in the lung that persists until the day of challenge. So here are the results of the flu H1N1 challenge. These are viral loads in bronchoalveolar lavage. Again, if we go counterclockwise, in the sham controls, we see the expected viral load course of about 7 logs of virus that's controlled in about 10 days. The clinically licensed vaccine, the inactivated flu vaccine and the flumis vaccine, resulted in about a log reduction of these, but not very impressive. We see several log improvement of protection with both the mRNA vaccine and the RAD52 vaccine given intramuscularly, a slight further improvement with the RAD52 vaccine given intranasally, and a substantial improvement given the RAD52 vaccine intratracheally with only about 3 logs of peak virus that's gone by day 4. If we look at viral loads in nasal swabs, then we see a very similar hierarchy. In the sham controls, there's about 7 logs of virus that's controlled by day 14. This is slightly reduced by the clinically licensed vaccines, the QIV and the flumis vaccine. It's further reduced by the mRNA and the RAD52 vaccine given intramuscularly. And that's even further reduced when the RAD52 vaccine is given intranasally or intratracheally. And with the intratracheal vaccine, we only really see 1 day of virus on day 1, and it's gone in the majority of animals by day 2. So a very similar profile that we see with flu as we did with COVID. If we look at correlates of protection with the clinically licensed vaccines, the inactivated flu vaccine and the flumis vaccine, then we look at a variety of immune parameters in this heat map compared with which parameters correlate with day 2 viral loads in BAL, day 2 viral loads in nasal swabs, the time to virologic control in BAL, and the time to virologic control in nasal swabs. Then it's not a surprise that the correlates of protection for these vaccines essentially are serum antibodies. The reason why I say that's not surprising is because that's really the only responses these vaccines generated. They did not generate cellular responses, and they did not generate mucosal responses. So it's not a surprise that serum antibodies correlate with protection. If we look at all the vaccines, then we see a much more rich profile of correlates in which if we look at all the vaccines put together, both serum and mucosal antibodies as well as mucosal T cell responses are all correlated with the protection. Interestingly, peripheral T cell responses really did not correlate with protection, which is sort of the hole here, and we're trying to understand that better. But it is clear that in this study, serum and mucosal antibodies as well as mucosal CD8 and CD4 T cell responses correlated with the protection. So my last short story of my talk is about the pandemic threat of H5N1 avian influenza. As I'm sure people in this audience know, there's an outbreak of H5N1 avian influenza, clade 2344B, in dairy, cattle, and poultry. About 70 cases are detected in humans to date. Most are clinically mild, but there have been several severe and lethal cases. The virus in cows has bovine adaptation mutations and has not yet acquired the capacity for person-to-person transmission. We certainly hope it never does, but I think most people agree that this does represent a pandemic threat for humans should the virus acquire those mutations necessary for a high level of human infectivity and transmissibility. So our research objectives for the H5N1 projects are to develop, or were to develop an H5N1 model in non-human primates with a current outbreak strain, and then to evaluate vaccine candidates, antibodies, and therapeutics. So our modeling paper is a model in cynomolgus macaques, and I'm just going to show this one slide. We have a detailed manuscript that's going to be published soon on the development of this model, but this is a clinically significant model in which there's a high degree of mortality. We infected 24 cynomolgus macaques with 4 different doses of the H5N1 challenge virus from the Texas isolate, and all 24 out of 24 animals died between day 4 and 7 of a rapidly progressive pneumonia that was proven by PET scanning and histopathology as well as virologic measures. And you can see this is a rapidly clinically progressive and rapidly lethal disease. The vaccine study was a study that we did in 24 cynomolgus macaques, in which we produced in-house an mRNA vaccine and a Rad52 vaccine expressing the H5 antigen from the Texas isolate. Group 1 was the mRNA vaccine given intramuscularly. Group 2 was the Rad52 vaccine given intramuscularly. And Group 3 was the Rad52 vaccine primed intramuscularly and boosted intratracheally. And then at week 16, so the boost was at week 8, and at week 16 we challenged with 10 to the 6 TCID50 of the H5N1 Texas isolate. These are serum antibody responses, so serum IgG responses by ELISA to the H5 antigen. And you can see that responses are primed after a single dose and were boosted in all 3 groups after week 8. And so you can see at week 10 there's a substantial increase in serum antibody responses. And the antibody responses were really quite similar between the mRNA platform and the Rad52 platform. And interestingly, the boost of Rad52 intratracheally led to a similar level of serum antibody titers as the intramuscular boost. So you don't lose anything by boosting mucosally. You actually get the same serum responses, if that's what you think is important. You actually get the same serum responses by boosting mucosally compared with boosting intramuscularly. But if you look at mucosal antibodies, then again we see this black and white difference, that there's a substantial improvement in mucosal IgG in the bronchoalveolar lavage in the group that received the intratracheal boost. And if you look at mucosal IgA, it's a black and white difference that you only get mucosal IgA if you boost intratracheally. You do not get mucosal IgA by either mRNA or AD platforms given intramuscularly. If you look at CD8 T-cell responses, then we get CD8 T-cell responses with both the mRNA and the Rad52 platforms in peripheral blood on the top, slightly higher with the AD platform than the mRNA platform, which is as expected. But if you look at CD8 T-cell responses in bronchoalveolar lavage, if you look at the mucosal CD8 T-cell responses, and these are resident memory CD4 and CD8 T-cell responses on the bottom, you only get these responses with the intratracheal boost, and you do not get resident memory mucosal T-cell responses by intramuscular boosting with either mRNA or AD vectors. The protection against mortality was 100% with all 3 vaccines. So in the sham control group, 5 out of 6 animals died by day 7, very similar to our model building study. 1 of the sham controls survived in this case, but 18 out of 18 vaccinated animals all showed 100% protection against mortality. Differences between these groups was apparent if we look at viral loads, and here we look at viral loads in plasma, BAL, and nasal swabs for the 3 different vaccine modalities as well as the sham controls. Unlike the H1N1, in which there was no viremia, then we actually did detect plasma viremia in all of the sham control animals for H5N1. So it's just a much more pathogenic infection. And all 3 vaccine modalities blocked plasma viremia. If we look at virus in the bronchoalveolar lavage and nasal swabs, then the mRNA and the Rad52 vaccines given intramuscularly look very similar, and we see about a 2-log reduction of viral loads in the BAL and nasal swabs. But if we use the Rad52 intratracheally, we see a stunning increase in protection of about 5 logs in BAL and about 4 logs in nasal swabs. And just to emphasize this point, then if you look at the reduction of viral loads on day 2 in BAL, then compared to sham controls, then the mRNA and the Rad52 give about a 2-log reduction of viral loads, and we see a 6-log reduction of viral loads if we boost intratracheally. In nasal swabs, we see about a 2 to 3 log reduction with the mRNA and the Rad52 given intramuscularly, and about a 5 log reduction given intratracheally. So we do see this substantial delta uptick of protection when we can induce mucosal immunity. So my conclusions for influenza: both mRNA and Rad52 provide superior protection compared with QIV and FluMist in the NHP H1N1 influenza challenge model. The immune correlates of protection for the clinically licensed vaccines are serum antibody titers, which is not a surprise because that's the only responses that these vaccines induce. In contrast, the mRNA and the Rad52 vaccines induce robust antibody and T-cell responses, and both correlate with the superior protection observed. But the Rad52 mucosal boosting induces comparable systemic peripheral immunity compared with intramuscular boosting, and the mucosal boosting substantially improves mucosal immunity, both mucosal humoral and mucosal cellular immunity, and this results in a substantial increase in protection in both the H1N1 and the H5N1 influenza challenge model in macaques. So I'll leave you with just a thought, and these data suggest a path for improving respiratory virus vaccines. Should future vaccines include not just a shot in the arm, but also a spray or a puff. So I'll leave you with that thought, and this is our group at Beth Israel Deaconess Medical Center and the lead authors of the various studies that I showed. I'll stop there, and if there's any time for questions, happy to take any questions from the audience. Thank you very much.
Thank you very much for presenting such excellent and insightful findings on the importance of mucosal booster vaccine against respiratory viruses. Now move to the Q&A session for both speakers. Yeah. So questions from the floor? Okay.
My question goes to you. It was very interesting that the intranasal Ad52 was much more efficacious compared with FluMist. So do you have any explanation? They are all live viruses.
So the FluMist vaccine has not shown very good immunogenicity in humans either, and I think that the thought is that it might be over-attenuated. Because, I mean, theoretically, a replicative live attenuated virus given mucosally should give substantial mucosal immunity. So the empiric observation that it doesn't is probably a factor of the degree of attenuation.
I hope you remember my previous question in Shanghai. I asked you why intratracheal was much better than intranasal. Here you have shown the TRM responses in bowel fluid. Could it be correlated with protection, superior protection of intratracheal?
Yeah, I think so. I think that the intratracheal administration was much better than the intranasal administration. I should emphasize that the way we did these experiments is we wanted to isolate the anatomic regions. So the intranasal administration was really just drops in the nose, and the intratracheal administration was just drops in the trachea. So in humans, then we think the intratracheal administration is very much clinically translatable because a more robust spray in the nose would naturally get vaccine beyond the nares and into the posterior pharynx and maybe even into the lungs. So the distinction between the intranasal and intratracheal is specific for macaques as a proof of concept study. But we do think that probably multiple different clinical translational applications could likely achieve the same mucosal immunogenicity as what we saw by the intratracheal administration in lungs. But it's clear that the lungs are a very dynamic inductive organ, and much more so than the nares. And so I think that in humans, then there would need to be phase 1 clinical trials that would probably compare mucosal immunogenicity of different modes of administration. And probably the further down a vaccine could get, probably the more immunogenic it would be.
Yeah. The TRMs in lung tissue is very notorious for its short duration of persistence compared with other tissues. So what do you think the ideal interval between prime and boost in human settings?
Yeah, it's a very interesting question. I think that's an empiric question. I don't think we know much about the biology of resident memory T cell responses in the lungs, but I think that that's an area that we really need to know more about, and particularly if the concept of mucosal immunity is going to be important moving forward. I think that there's just a lot we don't know about it. So I don't know what the optimal duration is. I'm not sure anyone does, but I think that that is a cell population that needs to be studied in more detail, ideally in humans, and then if that's not possible, then in animal models.
Thank you.
Any? Okay, Gongri.
My name is Kang Ho Lee. I have two questions to Martina Oaks. First of all, regarding the manufacturing, you give us the information through the slide. Manufacturing company is a small number in the slide. So when I was the deputy minister in the Ministry of Health and Welfare during the COVID-19, I had informed that many factories has been designated as the factoring company by CEPI. But on the slide you presented is a small number of factories. So why the number is small? And then according to African CDC, Africa wanna produce vaccine 60% of the African continent by 2040. So if the African countries build a new vaccine factory, in that case, CEPI is going to consider the factory platform in Africa. Second question is regarding the patents.
Regarding
Patent. So within the when take place is the pandemic, within you can provide vaccines, right to vaccines to any countries. So you have some problems on patents. During the COVID-19, we used the patents through the MPP organization, but it takes too much time. So how do you solve the patents problems within 100 days? Thank you.
Yeah, these are excellent questions. So the first one I think is easier. The 4 manufacturing facilities that I mentioned, they are a part of what CEPI calls the Vaccine Manufacturing Facilities Network, which is facilities that we onboarded through a call for proposal. And these are facilities where we are investing into building up their capacity. So this is not product-driven, it is really facility-driven. And the ones I mentioned, the 4 are facilities where we are investing in RNA technology. We have other facilities where we are investing in other technologies. They are not necessarily all the developers that we work with that actually have the ability to really manufacture. But they are facilities where they're all in LMICs, where we are investing particularly in building up the facilities' capacities, product agnostic. It's more technology driven. I hope that clarifies that a little bit. And on the question about the intellectual property, CEPI is actually not licensing or owning intellectual property. The intellectual property is taken care of by the developers, by the partners we work with. So if you have a platform for which you have all the technologies, then you would be the owner of the technology. If you have a technology for which you do not have full ownership, then you are expected to license the intellectual property that you do not own yourself. And CEPI is not either funding intellectual property or owning intellectual property. That you means that CEPI is supporting the licenses. Is that your question?
Yes, the question is the patents, intellectual property patents, but CEPI is not solve the patent problems. So what's the role of CEPI at the time?
I think in intellectual property questions, we have to rely on the developer, and that the developer is working with other entities that are more specialized in this, particularly the Medicines Patent Pool, for example, does make license arrangements if they can succeed to make them. But CEPI, we are intellectually as well not equipped to deal with intellectual property. It is such a complicated and broad field. We don't have the specialty trained people.
Any more questions? Okay. I think.
Thank you. My question is Dr. Baroj. So my name is Vishnu. I come from a company called as SPI Pharma, based out of USA, involved into the manufacturing of vaccine adjuvants like alum, sustainable saponins, and synthetic DLR agonist. So I wanted to ask, like, how's your experience around using any vaccine adjuvants for intranasal or mucosal influenza vaccines?
So we have some ongoing studies where we're looking at some protein nanoparticle vaccines with adjuvants given systemically versus mucosally. Those studies are earlier along, and so we don't have any results yet, but we'd certainly be interested in, I mean, I showed data on mRNA and vector-based vaccines, and we'd certainly be interested in looking at maybe working with partners on looking at other platforms, including adjuvants. But as of now, we don't have a lot of data ourselves on mucosal adjuvants. There are, in the literature, there are some mucosal adjuvants that appear to be designed specifically for mucosal use. I think a rigorous head-to-head study of different adjuvants, I don't think has been done for mucosal application. But I mean, one of the key things is the mucosal immunotechnology. So we spent a long time trying to develop and then perfect mucosal immunogenicity assays, both for mucosal antibody responses and mucosal T-cell responses. And the ability to quantitate mucosal immune responses in a highly reliable way, I think is a critical advance for looking at these mucosal questions.
Just to add, if I may, so our partner in Inimin Corporation, based out of Montana, has developed one synthetic TLR4 ligand, which showed fantastic lethal, protection for the influenza for both the H1N1 as well as H7N1. So we'll be happy to connect and discuss. Thank you.
Happy to talk. Thank you.
Thank you very much. Because of the time limitation, I think it's to ask you to ask a simple question and okay.
Okay, I have a question to Professor Dan Barrow. I'm Dokeun Kim from Institute Pasteur Korea. So you answered to the previous question about the difference of intranasal and intratracheal vaccination. And you mostly mentioned about some technical issues about the administration. I wonder if there is any immunological difference between intranasal and intratracheal administration, like some different lymph node drainage or something others?
Yeah, I think so. Again, it's an area that I think really needs to be explored more, but the basic biology of the mucosal immune system is really, our knowledge is really in its infancy. We don't really know where immune responses are induced or boosted with a mucosal antigen. It's clear it's not just an overlap with systemic immunity, because we see these drastic differences, such as intramuscular administration gives huge serum antibody titers that really don't result in mucosal antibody titers at all in terms of mucosal IgA. So there is a large distinction between the mucosal immune system and the systemic immune system. And we don't really know where the inductive sites are, but certainly if you look histologically, there's a whole lot of lymphoid tissue in the lungs. In the BALT, the bronchial associated lymphoid tissue, as well as in the posterior nasopharynx, and much more so than in the nose itself. So I think a hypothesis, which is not really proven, but a hypothesis is that you really want to get mucosal vaccines to the areas of the respiratory tract that have the most lymphoid tissue associated with them. So that's our going hypothesis, but it's definitely not proven.
Okay, thank you very much. There are a lot more questions, but unfortunately, the organizer asked me to stop. So, yeah, I think we need to have a lunch now. So, I think please give a big round of applause for both speakers. I think. Thank you very much.
Thank you to all of our morning speakers and for all of the excellent questions from the audience. We will now serve lunch outside, and we will meet back here for the afternoon session starting at 1:30 p.m. So enjoy your lunch. Thank you. Okay, speakers, distinguished guests, welcome back to the Global Vaccine Forum. Thank you for finding your seats. We'll now head into session 2, Tackling Challenges in mRNA Vaccine Development. It is a great pleasure to invite Professor Jun-Heng Ri of Jeonnam University Medical School to chair this session. Professor Ri is a distinguished expert in molecular microbial pathogenesis and vaccine biology. His pioneering work on Vibrio vulnificus led to the first full genome sequencing of the bacterium and the identification of the deadly RTX toxin responsible for its virulence. His team developed a vaccine and therapeutic monoclonal antibody targeting this toxin. While studying V. vulnificus in the 1990s, he discovered the flagellin protein's strong mucosal adjuvant properties, later linked to TLR5 signaling. His research demonstrated that TLR5 remains functional in aged animals, suggesting flagellin's potential as a vaccine adjuvant for the elderly. Since flagellin has been used as an adjuvant in vaccines against bacterial, viral, and non-infectious diseases like cancer, allergies, and Alzheimer's. Professor Ri served as president of the Korean Vaccine Society from 2013 to 2015 and the Korean Society for Microbiology in 2022. He currently directs 2 research centers at Jeonnam National University and has authored over 180 scientific papers and holds over 50 patents. Please help me welcome Professor Ri.
Thank you. Thank you for the lengthy, introduction. That's too much. Welcome to the, session 2. In this session, we are going to, deal with very fashionable science, AI biology and structural biology. We have 4 speakers. The first speaker will be Dr. Min-Kyung Park. She's a soaring star in the field of AI biology, and she significantly contributed to the David Baker's Nobel Prize last year. Her first authored science paper dealing with Rosetta fold is very much cited by all over the world. And her technology became the essential tool for future vaccine design. She's going to present transforming vaccine development with AI. Dr. Beck.
Okay. Thanks for the kind introduction. Okay. Hello, I'm Min-Kyung Baek from Seoul National University. And today I'll talk about how artificial intelligence is transforming vaccine development, particularly through the structure-based strategies that allow us to rationally design vaccine antigens. Then why structure-based strategies are important? our immune system, especially the B-cell mediated immune system, doesn't recognize linear sequences. It actually recognizes the three-dimensional structures. Neutralizing antibodies actually target specific conformations of viral proteins, especially in their pre-fusion state. And that's why the Professor James McLaurin actually developed the COVID vaccines by introducing some mutations that can stabilize the pre-fusion state so that our immune system can prepare for the viral infection. So by understanding and engineering these structures, we can design antigens that are more precise, more immunogenic, and more stable, and also more predictable in some protective responses. As almost of you already know, in 2024, the last year, the Nobel Prize in Chemistry was awarded for breakthroughs in AI-based protein structure prediction and design. So tools like AlphaFold and RosettaFold revolutionize the structural biology by enabling highly accurate protein structure prediction directly from sequences. So as shown here, on the left, it shows the AlphaFold success and how AlphaFold can predict the structures in high accuracy. So now those AI methods are actually make it possible to predict 3D structures from sequence, not solely relies on the cryo-EM structure or X-ray crystallographies to get 3D structures of our target antigens. On the right, I shows the RF diffusion and protein MPNN, the design structure or designed proteins based on those methods. This AI-based protein design method enables us the efficient protein design. So thanks to the work from the Google DeepMind and also from the Baker Lab in the University of Washington, we are now having AI tools that can predict the protein structures in high accuracy and also design proteins with desired function. So those high accuracy protein structure prediction, like AlphaFold2 method, actually makes us predict the conformational B-cell epitopes more feasible. So I bring some examples from the different groups. The first one is a DiscoTops 3.0, which basically tries to improve B-cell epitope prediction ability by using the predicted structures from AlphaFold and redesigning the sequences and get some representations from there. And the other work from the other group, which published in Bioinformatics, this method also utilized the predicted structures from the AlphaFold to predict the conformational B-cell epitopes. So like AlphaFold and RosettaFold, the protein structure prediction AI method, they are enabled us to predict the B-cell epitopes, not only the linear epitopes, but also the conformational epitopes. But the main limitation at this area is because of the lack of training data. The ability of those AI methods still has some limitations, especially for the new antigens haven't been studied, fully studied in our field. And I'd like to introduce how we can utilize the protein structure prediction and protein design tools to stabilize the vaccine antigens and how we can utilize those tools to rationally design vaccine antigens. So here I showed you the two conformations of the spike protein. One is the pre-fusion state and the other is post-fusion state. So for the spike protein in COVID, the SARS-CoV-2 virus, it is well known that the spike protein actually undergoes the conformational change upon fusion to the cell or the host cells. So it's really important to stabilize the pre-fusion form to make a vaccine efficient. So the Professor James McLaurin actually introduced the two proline mutations to stabilize the pre-fusion state by analyzing the experimentally determined structures of those pre-fusion and post-fusion states. You may have why he introduced the you may have a questions why he introduced the proline to stabilize the pre-fusion form. So I just bring some chemical structures of the proline. The proline, because of the unique nature of the side chains, proline doesn't have backbone hydrogens, which makes it hard to form any stable secondary structure like alpha helices or beta strand. And that's why Professor J.S. McLaurin actually tries to put two prolines in pre-fusion state, in the loop regions of the pre-fusion state, which actually changes to the alpha helices in post-fusion state. And this mutation actually stabilizes the pre-fusion. So I have a question that whether we can find these mutations with AI. So Professor J.S. McLaurin utilized his insight to introduce those mutations, but if we can do that by AI, then it will be much better or much faster to design the vaccine antigens. So to do that, I utilized the protein MPNN, the AI method, which basically designing amino acid that can stabilize the given the 3D structure. So this protein MPNN is developed by Eustace in David Baker's group. Now he's joined the Gyro Therapeutics, the spin-off from the Baker group. And this protein MPNN actually take the protein 3D structures as input and tries to generate the sequences that can have the provided input structure. And one good thing of protein MPNN is not designing the sequence from the N terminal to C terminus. You can randomize the order of the amino acid that designed so that it can utilize the motif information, given the sequence information if we wanna put some, if we wanna just mutate few residues. So you may think if we apply the protein MPNN to stabilize the pre-fusion state, you may imagine some pipeline like this. Take the pre-fusion state of spike proteins from the PDB or predict them using the AlphaFold, then run the protein MPNN to mutate the amino acids so that we can stabilize the pre-fusion state, then get the optimized sequence. But one problem, what if the mutations interrupt the pre-fusion state form designed by protein MPNN also can make the post-fusion state more stable? Because we only consider the pre-fusion state of the spike proteins, it does not guarantee it will lock to the pre-fusion state. It may more stabilize the post-fusion state. To avoid this, in our group, the student in my group, the Junhyuk, he tries to develop the multi-state MPNN, basically takes both pre-fusion and post-fusion state to design the mutant sequences which selectively stabilize the pre-fusion form. So we basically tweak the protein MPNN a little bit for the inference mode. So it takes the both pre-fusion and post-fusion state and for each residue, it calculates the preference of the amino acids and pick what amino acid that preferred in pre-fusion form, but didn't preferred in the post-fusion form. In this way, we can selectively design a sequence which prefers pre-fusion but not post-fusion. So I'd like to show some showcase whether our method is more effective than just considering the pre-fusion state. So for the SARS-CoV-2 spike protein, we take the wild-type sequence and predict the 3D structures of this sequence. And when we just tries to design or tries to mutate some amino acid to stabilize the pre-fusion, when we consider pre-fusion state wholly, then it gives the two lysines. But when we take care of both states, it introduces the two proline as same as J.S. McLaurin's approach. So we did some in silico validation because we don't have, in our group it's only dry lab, so we first do some in silico validation using the AF2 rank, which published in PGX review letters in 2022, which can evaluate or estimate whether the given sequence is well suited for the given structures. So as shown here, the blue means the sequence fits well to the given structure, and the red means it may be destabilized because the sequence doesn't like the given structure. So in this case, the mutant sequence we designed by considering pre-fusion state only and by considering both states. Both mutants actually prefer the pre-fusion structures. But when we look into the post-fusion structure, as you can see here, the sequence designed by considering pre-fusion state only is still like the post-fusion structure. But the designs by considering both states actually disfavor the post-fusion state, which makes it much better to selectively stabilize the pre-fusion state. So based on this observation, we are trying to apply this multi-state antigen design for various viruses, including influenza, COVID-19, and Nipah, Hendra virus, chikungunya, and so on. So this work is done by collaboration with Professor Nam-hyuk Cho at the Seoul National University, and also the Center for Vaccine Research in Korean National Institute for Health. So because it's still undergoing project, I'm not going to share the result yet, but it shows some promising result. Our designs actually works stabilize the structure, and it shows some immune activity. And at the same time, there's also some needs to develop the method which can scaffold the multi-epitopes for the vaccine design. So some cases like the antigen structure cannot be stabilized by mutating some amino acid, or some cases like people want to try to reduce the size of unvaccinated antigens. In those cases, it may be better to designing a new protein that have the antigen epitopes, but in different scaffold. So there's some work published in bioRxiv, the preprint, from I think Bruno Correa's group, which named RF joint 2. So it takes the epitope information from the proteins. So it takes the epitope structure and sequences and tries to design a new backbone scaffold that can accommodate the given epitope structures. But in this method, it has some limitations. So users actually need to predefine where to put the epitope in the sequence. And also, it's really hard to see the slides, but also that it's difficult to also the scaffold the discontinuous epitopes, which basically some epitopes spatially close to each other, but sequentially in the different region. So we are trying to develop some AI method which tackle this limitation by automating the epitope positioning and also the geometry-aware integration of those epitopes to the design scaffold. So this work is currently done by the Jaekyung Lee, and I'd like to share some result once it's finished. So in today's talk, I mainly talk about the AI-based vaccine design pipeline, our ongoing research. So it actually utilized the protein structure prediction method like AlphaFold and RosettaFold to get a 3D structure of the viruses, especially for the multiple states of the viruses, like a pre-fusion and post-fusion state. And for the cases we want to just stabilize the pre-fusion form, then we can utilize the multi-state MPNN, which can selectively stabilize the desired state only. In some cases, if we want to reduce the size of the antigens, or we want to incorporate multiple epitopes from the different proteins, then we can utilize some backbone scaffolding AIs that can make antigen structure having different epitope structures on the surface. And I'd like to thank for all the collaborators who worked with me so far. And I'd like to also thank to all the group members, especially Junhyuk and Jaekyung, for their work on vaccine design AIs. And also I'd like to thank for all the funding resources. Thank you.
Thank you. Thank you for your excellent, fascinating presentation with ongoing projects. We are going to take questions at the end of this session. Okay. Please give her a big hand. Next speaker is Robert Paris, who is the Vice President at Moderna. He is in charge of therapeutic area and infectious diseases. He leads the development of non-respiratory vaccines in Moderna, such as cytomegalovirus, genital herpes, Epstein-Barr virus, norovirus, Lyme disease, and others.
So thank you for the kind invitation to speak at this event. So, I just celebrated, just a little over 5 years being at Moderna. I joined in the heat of the pandemic. And prior to that, I had been at GSK and had worked on self-amplifying mRNA vaccines. And that led me to my time at Moderna. And I will say I've been focused in the last 2 or 3 years on our non-respiratory portfolio, but I have been involved with clinical development of mRNA, 1273, 1283, as well as our flu vaccine. And I'm going to struggle with this a bit. So here are my disclosures. I do work for Moderna, and I do hold equities in the company. So I think we're going to hearing a lot about the mRNA platform technology as it enables vaccine development. But I think Moderna started out as a company which really was focused on delivering a diverse portfolio of products. I think initially we were really focused on cancer and other therapeutics. And so as a result of that, though, of the pandemic, we really made a wholesale transition almost to broaden our pipeline of infectious disease products. But I show this slide just to show that for the encoded mRNA, we can actually generate missing proteins. So for rare diseases, particularly metabolic disorders where you have infants affected by inherited deficiencies of enzymes that lead to propionic acidemia, methylmalonic acidemia. Those are really key considerations as we look at this as a platform for rare diseases, and perhaps even amenable once you have proof of concept in one rare disease to easily extend that to additional rare diseases. The other part of our portfolio is cancer vaccines and therapeutics, and perhaps many of you have heard of the individualized neoantigen therapy, which is really targeting taking someone's tumor, sequencing it, and developing a personalized vaccine, which is what we used to call it. However, it does work a little bit different, perhaps mechanistically from a traditional vaccine. We also have now advanced actually what we're calling a checkpoint vaccine into advanced development. And that checkpoint vaccine really it's a misnomer. It's actually, we're utilizing the immune response to PD-L1 and IDO in a way that actually it effectively blocks some of these immunostimulatory responses that we see are diminished in cancers. And so this is another way to have a more off-the-shelf type of approach to severe cancer. The other is that we've looked at a number of different delivery systems. And so for our rare disease portfolio targeting metabolic diseases, we use an intravenously administered formulation, a different LNP, a different lipid, different cationic lipid, to deliver that. And similarly, in a collaboration with Vertex Pharmaceuticals, who have treatment for cystic fibrosis, we now have a clinical trial ongoing where we're utilizing an inhaled nebulized version of a lipid nanoparticle to deliver that to the airways in patients with cystic fibrosis, which, if you've noticed that disease at all, has very thick secretions in the mucus, as well as colonization and biofilms. So I think this is hopefully we'll have a key readout in a not too distant future. As this is probably kind of the next wave of innovation in mRNA technology will really be around delivery systems and being able to utilize LNPs to target specific tissues. Our R&D strategy, as I mentioned, was really to build a pipeline, in those years around the pandemic, and we really took advantage of the revenues we received to actually build a diverse portfolio. We really spent a lot on R&D. In the 2023-2026 timeframe, we've looked to expand our respiratory vaccine portfolio. So we've licensed both an RSV. We have now 3 products approved to include Spikevax, mRESVIA, which is the RSV vaccine. And more recently, even with the change in leadership at HHS, we've received recent approval for our next generation COVID vaccine, mNEXSpike, or mRNA 1283. We also will be reviewing, we just have received also approval for an sBLA for RSV in high-risk adults from 18 to 59. And we've also have a combination vaccine, which we have not yet submitted the BLA. We actually pulled back that BLA because we wanted to, the FDA was asking for us to develop and provide efficacy data for a phase 3 trial. But we hope to have that licensed within the next couple of years. I'm going to focus a little bit, though, on our non-respiratory products today, really looking particularly at our latent virus programs. So we are planning to try and submit or have approved additional 5 different programs in rare diseases, oncology, as well as the latent. And then the 2 vaccines, which I won't talk only about one of them today, is CMV, which is in late-stage testing, norovirus being the other one. And I know that this has been a very common question we get from a lot of people because of the particular severity of this last year's norovirus season. So I think it's not lost on this audience that mRNA technology is really well suited to address immediate impact on long-term health effects, particularly the latent viruses. As we know that these latent virus characteristics, particularly the herpes viruses, hepatitis B, have this immediate impact on infection, both in terms of birth defects for potentially CMV, EBV, or not EBV, but others, as well as infectious mononucleosis. And then some of these can lead to long-term sequelae, and particularly for EBV, we recognize this as cancer and potentially autoimmune-associated conditions. So there's several features, I think, of mRNA vaccines and attributes, really that both antibody and T-cell responses are important for viral control. And really, mRNA vaccines have been shown in this audience is that mRNA, they do elicit strong antibody and T-cell responses. The other advantage of mRNA is we can target multiple cellular entry pathways for latent viruses. So we can encode multiple antigens and antigen complexes, and I'll show you one of those, thinking us back to the previous topic, which was quite divergent from mine, about how important antigen design and structure is to really developing a vaccine, even for mRNA. It's not necessarily sorry, Dr. Oaks, no, don't mean to insult you, but it's not always plug and play when you're talking about some of these complex antigens. And then we can actually, so as I mentioned, we can include these complex antigens in the proper configuration, conformation to elicit the type of response that we want to target. So we're currently developing vaccines against a number of latent viruses, and these are all primarily herpes viruses, which we have in the clinic. I will talk more in depth about our cytomegalovirus virus vaccine, which is in phase 3 testing, and then also our Epstein-Barr virus. We also have an HSV2 vaccine, which is in clinical testing, and this is targeting recurrent genital herpes. So that vaccine is being assessed in people who have recurrent herpes, and we're looking at both immunologic as well as clinical readouts to see if we see an early proof of concept there. VZV is sort of a model antigen prototype vaccine based on really the success of Shingrix. I would like to also sort of point out that all of these herpesviruses are quite different in their biology, the immune responses they elicit, and the immune evasion mechanisms. So while mRNA 1468, our VZV vaccine, is sort of a Me Too candidate, we do believe that there's potential for improvement, or in terms of delivery of vaccines more globally, for recurrent shingles. So I want to switch now to CMV. So perhaps many of you have not seen a congenital CMV case, but this is probably one of the leading causes of congenital viral infection and non-genetic causes of sensory neural hearing loss in the US and worldwide. It's often a major under-recognized cause of miscarriage, stillbirths, or preterm births, and there was really recently a mini autopsy study which was conducted in less developed countries, which really highlighted the importance of CMV in the perinatal period in terms of health outcomes. The annual birth prevalence of congenital CMV ranges from about 1 in 70 to 1 in 208 births, with about 1 in 200 births affected in the United States. And the economic and clinical impact is significant. It's in the US, we think this amounts to about $6 to $7 billion in annual healthcare costs, as well as the complication that management of congenital CMV is really challenging because there's very limited prevention opportunities, inconsistent screening, and there's a lack of good treatment. So as a result, the WHO, as well as our U.S. National Academies of Medicine, going back more than 25 years now, I really felt this was one of the highest priorities for vaccine development. Clinically, the challenging thing with CMV is only about 10 to 15% of those infants who are infected actually have evidence of symptoms, to include hearing loss, cognitive, at birth. Most of these can be very severe symptoms and even result in death, which occurs in about 5% of these infants. And about half develop some term or long-term disability. Primarily, these are cognitive effects, to include the hearing loss, as well as potentially developmental delay. Interestingly, about 85 to 90% are obviously asymptomatic at birth, but still 10 to 15% of them develop some long-term disability. In total, about 1 in 5 infants then with congenital CMV, symptomatic or asymptomatic, develop a long-term disability as a consequence of this infection. So what are we trying to do with our vaccine? Really, we have designed our current focus to prevent CMV infection during pregnancy to reduce congenital CMV. So the idea is we would vaccinate women of childbearing age or potential prior to them becoming pregnant. And really, this is to interrupt the causal chain leading to congenital infection. We know that CMV infection in the first trimester actually has the highest risk of congenital disease, which is intuitive in the sense similar to Zika, that this is a time when there's significant neurofetal development going on, and this results in the highest impacts in terms of health consequences. And this idea is to prevent then onward transmission to the fetus, and then reduce the frequency of congenital CMV. Hence, our focus for initial indication is prevention of CMV infection in women 16 to 40 years of age, regardless of CMV serostatus. In addition, looking at a clinical outcome of congenital infection or disease based on some of the epidemiology I just described to you is very challenging because it would require a very large sample size for a trial as this. It really limits the ability for us to develop, I think, a vaccine. But I think it's an important thing that the development of CMV vaccines will be a long-term endeavor over time. So getting back to a little bit of the structure, I mean, this is really one of some of the data that actually very much attracted me to Moderna, and that was the ability to take 5 mRNAs and then to encode the pentameric complex of CMV. the pentamer complex is expressed on the cell surface. It's required for CMV entry of multiple cell types. And to have this all these mRNAs, encoded mRNAs, form their individual proteins, self-assemble, get trafficked to the cell surface, and then expressed in the conformationally correct position to elicit a neutralizing antibody response was, I thought, very impressive. Again, we also, historically, many vaccines for CMV historically have included a glycoprotein B response. And we do have an mRNA which encodes a full-length glycoprotein B. And glycoprotein B is a common herpes virus protein which really mediates that fusion of the virus and host cell membrane entry. Again, we included these antigen selection, as well as the fact that there is some establishment for proof of concept for glycoprotein B showing that it does prevent infection in women of childbearing age. So we conducted a phase 2 dose selection study, and this is, I'm going to go over this very briefly because I'm going to show you some of the neutralizing antibody data from that study, where we randomized adults, 315 of them, 18 to 40, with about the majority of them being CMV seronegative. And they received 1 of 3 doses or placebo. We had a placebo control group. And then we actually had also a safety expansion stage where we had selected a dose based on part 1, then looked at additional safety and immunogenicity data for the selected dose, which we then advanced to our phase 3 trial. So the objectives of the study were safety and neutralizing antibody responses, as well as binding antibody responses. We've also generated T-cell data, which is also, I'm not going to report, but I think really I wanted to show you is that one of the criticisms of the mRNA technology has been, the reported lack of durability of the immune response. And I think that's complicated by the fact that when you're using a single-dose vaccine against a respiratory virus, where you have this long history of previous infection, a lot of imprinting, and/or rapid viral evolution, that makes it challenging to look at the durability of a vaccine. So in this case, this is the data for the CMV seronegative group. And on the top line, the blue line is the vaccine response, use it to an epithelial cell infection assay. So we use 2 assays typically for assessing our vaccine because the epithelial cell assay does in the typical cell lines used to assess CMV vaccines historically, they don't express the receptor for the pentameric complex. So we really utilize this assay as our primary assay. And here you can see a nice response, and we include a dotted line there to show you what is the geometric mean titer for the participants in this study who were CMV positive at entry. And really the phase 1/2 data represents a larger number of participants through 18 months, and they showed that you could see the stability leveling out at 18 months, and then we have the additional data just recently. This abstract was presented at the ESCMID meeting in Vienna. We showed that it's durable through 36 months. So this appears to be fairly stable, and we'll have ongoing study with additional data from this study as well as our phase 3. So I find this pretty compelling because the rationale for a CMV vaccine, there may be a long interval between someone receiving the vaccine and then subsequently becoming pregnant. And so the idea here was that you could vaccinate potentially women of childbearing age, perhaps as low as 12, 13, as they're going into adolescence. So this led to the design of our phase 3 trial, which is a placebo-controlled observer blind study. And here we randomized both CMV seronegative participants and about a 4-to-1 ratio to CMV seropositive participants. The participants that we enrolled over the age of 20, we expected them to have some direct exposure to CMV, potential CMV, in the home, socially, or occupationally, to at least 1 child under the age of 5. This has been shown to be one of the highest risks for developing infection, CMV infection. And really, the women who are seronegative, who have a toddler in daycare or toddler at home, are at a higher risk for developing, their children are a little bit higher risk of developing congenital CMV. Pregnancy was exclusionary, and then we tested women prior to each immunization. And then they were randomized to 1-to-1 to receive the dose of vaccine. And this is given the same schedule I just showed you on the previous slide of 0, 2, and 6 months. Follow-ups for about 30 months with about 24 months of follow-up for efficacy. So our objectives for the study are to determine efficacy looking at a seroconversion from a negative to positive CMV IgG 28 days after the third injection, as well as safety. Immunogenicity is similarly, we're looking at immunogenicity and binding antibody responses. In addition, we're looking really interestingly at CMV viral shedding, and I'll show you some data later for our EBV vaccine with respect to the effects on viral shedding. Both in the CMV seronegatives after they seroconvert, so we bring them in, and every 3 months we collect urine samples for detection of CMV DNA, as well as in our seropositive cohort. And the idea here is that we would like to see if we see an effect in the compartments that might be associated with transmission, both to the baby vertically as well as horizontally. And so we're starting at the beginning of the study, we're also collecting serial samples to assess the seropositive women, the potential impact of the vaccine on a virologic outcome. So the phase 3 trial is ongoing. We've completed enrollment. It's being run in 290 sites in 13 countries. About 7,500 women have been enrolled, about 6,000 of them seronegative and 1,500 seropositive. And overall, I'm not showing the data. This vaccine's actually been decently tolerated, well tolerated, regardless of CMV serostatus. We sort of see similar responses regardless of whether they're previously infected with CMV or not at the time of vaccination. We haven't seen any safety concerns, and we did have a DSMB review of the unblinded data, safety data, last December, and they came back with no comments on or concerns about the safety. In general, the vaccine has elicited really nice antibody-mediated immunogenicity that's exceeded levels observed with natural infection, as well as boosting responses in seropositives. We have I haven't mentioned it here, but we do have T-cell data, CD4 and CD8 specific T-cell responses, which we've now submitted in an abstract for the ID week coming in October. But we do see durable T-cell responses as well. And then we will be hopefully showing, hopefully by the end of the year, we will actually have some efficacy data for this study. So let me switch now to EBV, which is this is a program that I find fascinating. And because of a link of EBV infection to infection, not only to infectious mononucleosis, but now really pretty well defined for multiple sclerosis, as well as cancers. I think most of us know that EBV seroprevalence increases with age, really depends upon the geographic location where people live. Typically, less developed countries results in EBV infection and a very early age, whereas in more developed countries, we see it perhaps more commonly in the teenage years, and that's when we typically see infectious mononucleosis as a complication of that primary infection. So EBV seropositivity is actually almost uniformly associated and universally occurs in patients with multiple sclerosis. And EBV seronegative individuals who have a negligible risk of MS. There's a recent paper that was published in Science, actually now about almost 2 years ago, looking at longitudinal data from a DoD cohort, the Department of Defense, showing that there's a 32-fold increased risk in a prospective study developing MS following EBV infection or seroconversion. And then IM on top of that increased that risk by about threefold, twofold or threefold. And that's shown here on this. It's hard to point on this graph, but in the middle, in the top panel. In addition, I don't think it's probably commonly understood in this audience the association of EBVs and cancer. So we know that it's associated with post-transplant lymphoproliferative disorders, which is typically observed in transplant populations, obviously, but also nasopharyngeal carcinoma, as well as Hodgkin's and other lymphomas. If we looked at EBV by IM status, and this is some work we did, was that people who have IM also have an increased risk of malignancy, typically the most common being Hodgkin's within the first 5 years of having infectious mononucleosis. So how have we gone about trying to tackle this pervasive problem? I mean, this infection's been around with humans probably as long as humans. And so we actually developed 2 vaccine candidates, mRNA 1189, and this is a multivalent vaccine that's really targeting largely the lytic protein. So this virus exists in 2 phases, replication active phase, where lytic antigens are expressed, and many of these lytic antigens are responsible for viral cellular entry. And so we've included an isoform of GP350, I'll just leave that for now, as well as GH, GL, GP42, which are involved with B cell as well as epithelial cell entry. The primary indication we're pursuing with this 1189 vaccine is for the prevention of IM, and with potentially the prevention of infection as a potential upside. And then long term, we think that there's possibility for the prophylactic prevention of some of these long-term sequelae to include MS as well as cancers. On top of that, we're developing another vaccine, mRNA 1195, which includes these lytic antigens. But then we've also added 2 T-cell antigens that are expressed within the latent phase, the latency 0 phase of EBV, being EBNA3A and LMP2B. And the idea there is that we will use this strategy perhaps to tackle some of the immune dysregulation that's occurring by EBV in MS, and then potentially look at restoring robust immune control of both lytic and latent antigens infection through vaccinations population. The other potential target for this vaccine is PTLD, as I mentioned. And similarly, I should mention that CMV is also a major problem amongst immunocompromised hosts and transplant patients. And we actually have an ongoing study with Lindsay Baden at Dana-Farber looking at our CMV vaccine in stem cell transplant patients. So I'm not going to go into too much on the phase 1 design, but I want to just kind of switch to some of, and also for both these studies, these were both randomized controlled studies. We tested multiple dose levels for our 1195, our lytic and latent antigen vaccine. We actually tested 2 different formulations. And then just show some of the neutralizing antibody data through the peak after the 3rd dose. So this is B-cell neutralizing antibody titers. So this is typically the assay we use to look at the contribution of the glycoprotein GP220, these B-cell targets on neutralization. And in the EBV negatives, you will see it really takes 3 doses to establish a neutralizing antibody response. We see interestingly, in the EBV positive, we see very little activity, B-cell neutralizing activity at baseline, which is significantly and strongly boosted upon vaccination. Similarly for the GHGL, and we have actually epithelial cell neutralizing antibody data, which looks equally as good as this, where we see a huge increase in the binding antibody responses to these antigens on our EBV seronegative population, and we see significant full log boosting, 1 log boosting, plus in our EBV positives. The data I want to kind of focus, though, on is that in this study, we actually took participants and we measured the viral DNA in their saliva from the time of vaccination. And this is all of the EBV positive participants. Because of the limited sample size, we've combined them. But on the top graph, what you're seeing is the frequency of detection in the gray bar of EBV DNA in saliva in the placebo group, and in the blue bar, the frequency of detection of EBV DNA in the vaccine group. And almost immediately after vaccination, you start to see a decline in the frequency of detection of EBV in the saliva. And then going down to the bottom graph, we've taken now those participants who had detectable DNA and actually quantified it. And so even those breakthroughs that we're seeing, those people who we detect EBV DNA in their saliva, we see a quantitative impact on the amount of virus in their saliva. So I think for us, this was really compelling to then advance this because of the acceptable safety tolerability profile we observed. And I will also say that we have also generated T-cell data for both these vaccines, and I'm not showing that because it has not been published and not presented anywhere publicly yet. But similarly for the 1195, we've repeated this analysis on impact on viral shedding and show that we impact viral shedding with both vaccines. If you ask me what is the mechanism of this, I don't know at this point. Those analyses are ongoing, but I think this is something that is quite compelling for the advancement of this vaccine for some of the conditions I described earlier. So in light of that, we now have begun a phase 2 study in patients with MS, and this is the patients that we're including in this study are randomized, in this placebo-controlled study, are actually patients who were recently diagnosed and have opted not to go on any kind of disease-modifying therapy at this point. We look at this as both a safety, primarily a safety study, as well as a proof of concept study to see if we can safely administer this vaccine. So we're doing this in a dose escalation fashion with a DSMB. And we're also looking for potential outcomes to indicate any biomarker of potential efficacy here, including serial MRIs, where we're looking for new gadolinium-enhancing lesions, preventions of MS relapse, as well as disability outcome measures. So in summary, I think I've shown that mRNA technology has enabled a broad portfolio that includes secreted, intracellular, and now inhaled therapeutics for rare diseases and cancer in addition to vaccines. We have generated, I think, favorable data to advance vaccines for latent viral infections, particularly EBV. We also have clinical data for our HSV2 as well as VZV vaccines, which we should be getting either published soon and or presented at conferences, which have potential therapeutic and preventive applications for potentially autoimmune diseases like multiple sclerosis and cancer like PTLD. And then as I mentioned, we anticipate efficacy data for our CMV vaccine sometime by the end of 2025. I would also mention that this vaccine entered into the clinic in 2017. So it is one of our major first products that we've started before the pandemic. But I just wanted to leave you that impression that sometimes it still does take a long time to develop a vaccine. We haven't gone from 10 years into 1 year. It's not always applicable, and for these latent viruses, that's certainly the case. So anyway, I'll conclude with that, and I would like to just take a quick thank you, in addition to the conference organizers, to also thank our investigators, study site personnel, lab personnel, and most importantly, all of our trial participants. Take any questions. I guess we're waiting until the end of the session, correct?
Okay.
Okay, thank you.
We'll take questions at the end of this session. Thank you. Let's move on to next presentation. Martina Oakes. I don't have to introduce her. She is in charge of mRNA vaccine technology and viral pathogens at CEPI, and supports CEPI's 100-day mission. Please go ahead.
Thank you. So this presentation now is about mobile GMP manufacturing. And so what I want to do here is I give you a little bit an overview of how we think about this, align on what is mobile manufacturing, give you some examples of commercially available mobile cleanroom technologies, GXP-ready equipment and technologies, give you some thoughts on the operational aspects that need to be thought about if you want to mobile manufacture GMP material, and then some conclusions. So first, really a little bit assumptions that we put into this presentation. The first is that GMP facility, that you will need a GMP-based facility with a fixed address where you will house manufacturing support functions such as warehousing, QC, QA, training, archives, data centers, and so on and so on. And we as well assume that the mobile manufacturing are standalone mobile units. So units that don't need a shell building in which they are deposited, but that the infrastructure will be as well associated with the mobile unit. You will need as well trained and skilled personnel, which most likely will have to travel with a mobile facility if you want to move it. And you will need a platform or process which has the freedom to operate. So that means it has to be at the destination of your mobile unit, the IP, the legal, the regulatory licenses, including health and safety permits. The host site needs to be as well GMP licensed so that you can manufacture your investigational product, including the capabilities and outsourcing for QC testing. You want to produce your investigational vaccine. That means you want to have as an output a labeled and packed vaccine that is suitable for clinical trial use. And you want to have this used for rapid local outbreak response only, probably, and that means you need to think about how to keep this facility warm in case, in the intermediate period between outbreaks. So prefabricated modules already exist that you can, where you have a prefabricated module that you put into an existing shell building, and they generally come at a higher cost than if you have an in situ construction, but they're usually faster to be installed than if you construct from in situ, but they are not necessarily designed to be routinely moved. And examples of those as, for example, the KU BioBox from Cytiva, or you probably are all familiar with the BioNTech from BioNTech. Mobile clean rooms as well exist. Probably the one you all have heard about is germ-free, and they as well usually come at a higher cost than if you construct from in vitro. They are designed to be moved as a standalone, and they are usually used for hospital pharmacy compounding or advanced therapy medicinal product generation or antibody therapy clinical phase manufacturing. GXP-ready process technologies, there are some existing ones. None of this has been approved in health products yet, but worth to mention is the microfluidics-based equipment. Centillion, Quantum, Micropore, and RPI MIT have developed such technologies, and these are just examples. I don't want to say these are the best or the only ones, by no means. These are just examples. As well of interest is the cell-free manufacturing technology for synthetic DNA templates. Licensed for GMP production is, for example, the doggy-borne DNA from Touchlight, or the synthetic DNA adapter technology from Syngoi. But you still have to think about hurdles to use such equipment for GMP production. And process qualification for the licensed products needs to be considered. Extractable leachables assessment needs to be done, and you need to secure a robust supply chain for the consumables and the reagents. Just an example, another mobile unit is being considered. It's, as far as I know, it's not used yet as such. It's not really ready yet, but it's under development. It's by Centillion. They call it a biofoundry in a box. And they need linearized DNA as input. Then there is a continuous flow IVT purification formulation. Capping is during IVT. Poly-A is included in the template, and it has a relatively small footprint with a relatively fast cycle time. So mobile production processes should be end-to-end. That means your starting material and your finished product should be made in the mobile facility. Otherwise, your mobile production will be dependent on input and output that is localized somewhere different. In-process and release testing, you should also have in your unit. Otherwise, you're dependent on off-site testing. And many of these hurdles for compact, lower volume GMP manufacturing are actually solved by the advanced therapies and medicines product sector, such as, I put here as an example, the fill finish, the AT closed vial technology, where you have a pre-sterilized vial and you inject your drug product through a stopper that then closes itself again through its elasticity. But there are lots of things you need to consider from an operational perspective for mobile manufacturing. And we just put here some of those considerations on the slides. So for example, workforce, recruitment and retention of trained GMP operating workforce that is willing to travel in the areas of need and short-term periods. And again, when I say that this is from an outbreak perspective looked at, that you want to have a mobile unit to be able to respond to an outbreak where it happens. The input materials, you need to think about how you will handle your raw materials and consumables, how this will be done locally in a GMP compliant, secured, controlled temperature storage environment, quarantines, testing, release, etc. You will have to think about how do you do all this. The local site infrastructure and utilities, including waste, need to be considered. If both the central base and your local site will require reliable, compatible utilities supplies for water, gases, steam, power. They will also need a security control and the necessary operating permits to handle, for example, flammables or controlled high purity ethanol used for the mRNA process and the sanitization. For qualification and validation, you need to think about the maintenance requirements of your equipment, and this may have to be happening more often. You may have a higher demand for validation and calibration if you actually move your equipment. And the equipment selection and the initial IOQ should include elements usually covered in the shipping validation. On QC and output materials, storage and logistics such, where will you do in-process and lot release testing? Will most samples need to be sent to the base site for testing or to other qualified laboratories for independent vaccine batch certification and release? You need to consider that. The GMP licensing, quality management and compliance. GMP and operating licenses are usually associated with a fixed address, and regulatory authority or law changes may be actually needed in order to accommodate for mobile production elements. How do you retain, record, and store your data? So your QMS, deviation, change control, training, batch records, and so on need to be fully digitalized. and that should be at both sides, your base site and your mobile site. And in terms of qualification and validation, you need to secure your archives for the samples and the records that will be required. Both base and mobile facilities will need reliable systems to ensure the data integrity, the network connection, data backup, and cybersecurity. And just to conclude, to realize mobile GMP production, legislative changes and regulatory harmonization may be required to rapidly expand a host site's GMP production licenses to encompass the vaccine, the investigational vaccine process. And you may need a dialogue with your regulatory agencies, not just the Korean FDA, but any regulatory agency that's then applicable at the potential host site as well. Although it's probably more rapid to establish than to build something in situ, the operational and the capital costs will probably be higher for your mobile manufacturing. And because of supply chain logistics, quality assurance and control, that can be as well more challenging than if you have a fixed site, and that may trigger higher cost of goods, and you have to think whether that is actually acceptable. The operational sustainability, you should think about what are you going to do with your mobile unit, and while there is maybe not a need for a localized manufacturing, and whether there's any products that you want to produce in there, and if there, during outbreak needs, and whether there's sufficient market for it. So mobile, as I said, mobile clean room and process technologies already exist, but you will have to think about hurdles that you need to overcome when you want to implement it, and those are mainly related to logistics and GMP compliance. Until your operations need to be considered, storage, cleaning, testing, archiving, etc. And you should think, is a truly mobile facility needed, or is prefabricated modules that are once moved and then left in place, like, for example, the underdeveloped under the US ARPA-H program, are those ones sufficient? Because a relocation may require as well a revalidation, site license changes, etc. And I need to acknowledge that my colleagues Rana and Emmanuel, Sepi, and in general the REMC colleagues made this presentation. It was spearheaded by them, and I'm here presenting to you. Thank you very much.
Thank you. Thank you for introducing CEPI's endeavor for so-called vaccine equity all over the world. We'll take questions at the end of this session. We'll have next speaker from IVI, Dr. Sang-Wan Seo. He's in charge of immunoassays in IVI and participated in the development of vaccine against hepatitis A and adenovirus 55. Dr. Seo.
Thank you for kind introduction. Good afternoon, everyone. Today I would like to introduce our latest initiative, the DKVAX project, which represents for the decentralized and accelerated mRNA vaccine production system. This project is designed to address critical global gaps in pandemic response and vaccine access. The COVID-19 pandemic revealed both the potential and limitation of mRNA vaccine. So while we witnessed their rapid development, we also saw that the production remained centralized, inflexible, and costly. The current mRNA vaccine production system uses a batch process in a fixed manufacturing facility, which limits the scalability, increase cost, and slow delivery, especially in low and middle-income countries. So recent efforts such as the BioNTech's BioNTainer and Quantum's Infinity platform introduced modular mRNA manufacturing solutions. So these systems mark important progress by allowing deployment in resource-limited setting and enabling regional production. But these systems still are based on the batch-based workflow, limiting efficiency and scalability. So we asked what next generation would look like. The conventional batch-based mRNA vaccine production is simple to implement, but limited by the reagent depletion and byproduct buildup, and it also suffers from batch-to-batch variation. In contrast, the microfluidic-based continuous mRNA vaccine production can maintain the steady state by constantly supplying the reagent and removing byproduct. So this enables higher productivity, sustained enzyme activity, and improved consistency. While it requires more advanced system, but it can enhance the efficiency and lower production cost at scale. So this continuous mRNA vaccine production system can provide a practical solution to overcome the limitation of current batch-based mRNA vaccine production system, making it a process for scalable and reliable mRNA vaccine manufacturing. So the objective of our DKVax is to develop, establish the third generation mRNA vaccine production system capable of producing 1 million doses of mRNA vaccine within 100 days. The DKVax is microfluidic-based, fully integrated and containerized system for continuous flow mRNA vaccine production. This system enables precise control, maximize the efficiency, enable flexible production, and also reduces the risk of contamination by allowing the single-use system. Now let's look at the two key modules of our system. On the left, the IVT module can constantly produce mRNA by mixing the template DNA and RNA polymerase in the microfluidic system. It uses precise flow at temperature control with integrated pump and touch panel for automation. Its compact design allows the scalability. On the right, the mRNA purification module uses a tangential flow filtration system to purify the mRNA as it flows through the system. This system ensures consistent and high purity mRNA output. Now we are testing the prototype machine. Now let's look at the next two key steps, LNP encapsulation and mRNA LNP purification. Our collaborator, InventJet, has already developed a GMP-grade LNP encapsulation system, which installed at a Korea vaccine company. For this project, they are enhancing the system by redesigning the LNP encapsulation cartridge to improve the product yield and ensure consistent LNP quality. After encapsulation, the mRNA LNP undergo purification using tangential flow filtration system. Together, these modules enable efficient encapsulation and purification of mRNA in a continuous and closed-loop process, supporting high-quality and scalable mRNA vaccine manufacturing. All modules will be fully integrated and installed within container, creating a ready-to-produce system capable of early mRNA vaccine production in the event of pandemic. And the container is designed according to international standard specification, enabling convenient transport and global deployment. This modular design supports scalability. Additional units can be deployed based on the vaccine demand. To maximize the space efficiency for experimentation, the refrigerators and defrigerers will be installed under the workbenches. To provide clear understanding of this integrated system, we made an animation, so let's look at this animation for a while. DKBX project has 3 steps. The step 1 is establishment of each module until end of this year. Step 2 is integrating the system and installed within container. And step 3 is full performance evaluation. The DKBX system will be evaluated using this kind of scenario to assess its ability to produce 1 million doses of mRNA vaccine within 100 days following initial outbreak. The DKBX system is not a standalone effort. It is supported by the IVI's broader innovation pipeline in mRNA vaccine development. So now we are collaborating with partners to develop mRNA vaccines against Rift Valley fever virus, MERS-CoV, Lassa fever virus, and COVID-19. And we are also innovating the mRNA vaccine platform by adapting the novel delivery system and UTR modification. The Lassa mRNA vaccine development project was funded by the Korea mRNA Vaccine Initiative. We will collaborate with SK Bioscience. The goal of this project was to develop rapid response mRNA vaccine platform for Lassa fever virus, which is one of the main cause for viral hemorrhagic fever in West Africa with relatively high morbidity and mortality. The vaccine candidates were evaluated in mice in terms of the antibody response and cellular immune response. The development of Rift Valley fever mRNA vaccine is supported by the Swedish government and CEPI. We are now collaborating with Aprigen, and Aprigen is now making the vaccine candidate, and IVI will evaluate its immunogenicity using the established immunological assays. This project is to develop MERS vaccine using a self-amplifying mRNA vaccine platform. This project is supported by the Swedish government. In collaboration with Professor Gerald McLeary at Karolinska Institute, now we are secure the vaccine candidate, and we will evaluate its immunogenicity and the protective efficacy in established mice model. To enhance the immunogenicity of mRNA vaccine platform, we are now collaborating with Professor Nelly Kim at Seoul National University. For this project, we are now using the influenza mRNA vaccine model as test system. The one of critical limitation of current mRNA vaccine is requirement for cold chain, which limit the distribution in resource-limited setting. So to overcome this, we are now testing the microneedle type mRNA vaccine in collaboration with Quad Medicine. This format not only improves the stability, but also eliminate the cold chain dependency. So now we are comparing the immunogenicity between the microneedle type mRNA vaccine and conventional delivery system with the goal of establishing more stable and user-friendly mRNA vaccine delivery platform. In collaboration with GC Biopharma, we are also developing the MERS vaccine. The goal of this project is to secure mRNA vaccine platform in South Korea as part of national preparedness strategy for future pandemic. So in summary, the DKBX system combined with prototype mRNA vaccine and platform innovation represent an integrated solution for future pandemic response. This approach builds a resilient and agile defense against emerging infectious disease. So continued collaboration and system integration, we aim to establish next generation mRNA vaccine ecosystem ready to respond within days, not months. I would like to thank many collaborators and funders to this effort in South Korea and globally. Thank you for your attention.
Thank you. Speakers, please come up to the podium. Hope IVI's decentralized mRNA vaccine production platform will meet the standard of CEPI and will come out soon. Yeah. Now, podium is open for questions and comments from the floor.
I have a question to Dr. Robert Paris. First, Thank you for the very interesting talk. I was definitely persuaded that the mRNA technique would be powerful against asymptomatic and latent viruses. And regarding this, I have two questions. And when it comes to those latent viruses, I think the immune memory would be particularly important. And of course, you have shown us that the neutralizing antibodies are persistent for more than 3 years. But I think evaluating the activity of memory B cells and T cells would be very important for those viruses. And yeah, I have a question for this. And second, I think one of the top interesting latent viruses would be HIV. And I think, it's just my personal curiosity, but I'm wondering if Moderna has a plan to expand your mRNA platform to HIV.
So I think the second question is quite easy to answer. And yes, we do. We have an HIV program that's in broad collaboration with IAVI and AID others. And actually, if you're familiar with Bill Schief and his work, we're approaching this through that gene line targeting approach where you're progressively mutating with vaccination towards a broadly monoclonal neutralizing antibody. So that work's ongoing. We're actually we had a bit of a setback with the government and NIH funding this year, but fortunately, we've had donors like Gates Foundation step up, so that approach will go forward, hopefully with the study by the end of the year in South Africa. That'll be the next study for that approach. We are not currently pursuing a therapeutic HIV vaccine approach for a number of reasons, but for prophylactic HIV vaccines, we still have an ongoing research collaboration. To answer your first question, so I didn't show the data, but we have generated data with memory T cell responses out to a year, and we see really nice, robust responses. I haven't shown that. We actually just submitted an abstract, particularly for specific to CMV. Similarly for EBV, we have not, but if you look at that slide, I showed you the impact on viral shedding. That effect is persisting for a year. So there's something active in the immune response, and we don't know if it's T cells or B cells at this point. We've undertaken some memory B cell work, and we have a paper in preparation now to look at this for RSV as well as flu. And I think, and also we've seen some of this work, we've done collaborations, not specific to the latent viruses yet, but we've done it for some of our respiratory vaccines. And we have some work being ongoing with Ali Elabedi, who's at Washington University, where we've done 5 needle aspirate studies for flu and COVID. And we really show increases in the long-lived plasma cell response in the bone marrow, but also in the memory B cell response to those antigens in the blood. So I think the data will be coming, but you're right. I think we're really feeling like T cell responses are going to be pretty important to this approach. I didn't show it, but one of the things that's really interesting, and I'll just add this as a side comment about mRNA technology, is that people maybe I'm anticipating a question is how do you guys expect to use a vaccine in patients potentially with patients with MS who are immunosuppressed? And there's been a couple papers now, one published in, I think, Immunity with Amit Bar-Or at University of Pennsylvania, as well as Arnaud Didier-Laurent at University of Geneva, where they've actually vaccinated with COVID vaccines patients who are on anti-CD20 therapies. And as you would expect, you don't see a B cell response, but you do see quite a robust T cell response. And actually, in their hands, the T cell response with CD4 and CD8 looks to be somewhat enhanced in those patients on the anti-CD20 therapy. And we don't really understand the mechanism of that. But we do see that these therapies in a sort of therapeutic setting, mRNA vaccination could be an add-on or additive or combination approach, kind of like what we're looking at with our INT vaccine. think I talked about that too much, but that's a collaboration with Merck, which is being used in combination with pembrolizumab, so a checkpoint inhibitor.
Okay, thank you.
Thank you so much.
Other questions? If not, I'd like to ask a question to, Dr. Peck. For your, multi-epitope, vaccine design, you may skew the, natural structure by, fusing different, epitopes. In our hands, if subtle change comes up, we see, good, IgG, responses, but, IgA responses goes down drastically. So do you have any tool or plan for, to predict more correctly what will be happening after, immunization?
That's a very good question. I think that's a kind of Nobel Prize award-winning question if I solve that. So it's more likely how we can understand for immunization process through some kind of modelings or some kind of the experimental researches. But at this moment, I'm not sure whether we can actually, predict what will be happen if we inject the designed vaccines to the patient, because we basically, at this moment, we don't fully understand how immunization actually works. So yeah, I hope we can find some answers in near future, but at this moment, it's pretty hard to predict, yeah.
So we still have, quite a big place where, the wet researchers could stand. Other question? Dan. Microphone.
This is a question for anyone on the panel. So can you contrast, in your view, what is the importance of having the manufacturing mobile versus having the vaccine itself mobile? In other words, room temperature stability, long-term stability. Do you need to have the GMP manufacturing facility mobile, or could you make it a fixed manufacturing facility in different parts of the world so there is democratization, but have the vaccine itself advances in formulation or otherwise so the vaccine is mobile? So what's more important, the manufacturing facility being mobile or the vaccine itself being mobile?
I'll go first because I think maybe Dan, you were maybe directing this at me a little bit because we didn't talk about that. I think, well, we've taken the approach, and I didn't mention this, that you can lyophilize mRNA vaccines, but that's not really a practical approach to improve stability, refrigerator temperature stability and distribution for a pandemic situation. So like our CMV vaccine that I presented, that's a lyophilized vaccine that has good refrigerator temperatures shelf life. So we've taken a little bit different approach. We've considered, and I think we've had a contract with BARDA for mobile manufacturing, but we have actually now established sort of regional specific manufacturing to perhaps decrease the distribution time. So I do think one component is you got to increase the stability, and that's going to take time. I think that's kind of, in a way, perhaps that's the next holy grail for mRNA vaccine, or next Nobel Prize is going to be in how you take modified mRNA and create something that's room temperature stable for a year or 2 years or whatever. I mean, some other mRNA technologies may be more amenable to that, but a modified seems to be a little bit more tricky. So we've taken a regional approach where we are now setting up, we have a manufacturing facility, hopefully will go online in Australia and Melbourne to serve the APAC region. And then we have additional capacity in Canada as well as in the UK now. We've set up a manufacturing facility just outside of Oxford in the Harwell Business Park. We did have some involvement with trying to set up manufacturing capacity in Africa, but there was a bit challenging. We ran into some challenges there. But it's not something we're taking off the table permanently.
Thank you. One more question. No? Oh, here. Last question. Please make it quick.
Thank you. I would like to maybe probe Dan's question a little bit further because I'm very curious to hear thoughts from Seppi, so Martina, and Dr. Saul from IVI. So what specific use case did you perhaps have in mind when you were envisioning mobile manufacturing units, especially considering that both the operational costs as well as the cost of goods are expected to be much higher? So I would love to hear your thoughts.
So from my perspective, what drives why you would think about mobile manufacturing is really that during COVID, certain populations were not really reached, and it might be easier to reach them if the vaccine is manufactured closer to where it is needed. You would eliminate long transport routes as well, although that is maybe not the major consideration. But you would, and vaccine uptake might be as well better if you actually produce more local. But please.
Yeah, actually, the mobile manufacturing system is early stage, actually. So, as Martina says, that currently is quite some high expense, but can be decreased the cost dramatically after in the near future. And so, as Dan's question, I also want to say that, as I already show in the conclusion slide, the platform innovation by improving the vaccine longevity and integration with that kind of the mobile facility can improve the vaccine inequity as shown in the COVID-19 pandemic. Usually we also show that. That's the answer.
Thank you. I'll close the session here. If you have further questions, you can ask them during the coffee break. Thank you.
Thank you, Session 3 Chair and speakers. We're going to break now for a brief coffee break. And so we'd like to ask you to be back in this room by about 3:15, 3:16, and then we'll get started for Session 3. Thank you. Okay, everyone, welcome back from the break. We'll now head into Session 3, titled Emerging Vaccine Platforms from Concept to Reality. This session will be chaired by Professor Jae-myun Lee at Yonsei University College of Medicine. Professor Jae-myun Lee is a distinguished professor and chair of the Department of Microbiology and Immunology at Yonsei. With over 2 decades of expertise in microbiology, immunology, and vaccine development, he serves as Deputy Director General of the Vaccine Innovative Technology Alliance, or VITAL, Korea, and is a member of IVI's Scientific Advisory Group. Professor Lee earned his MD and PhD from Yonsei University, followed by postdoctoral training in pharmacology at Johns Hopkins University School of Medicine. His research focuses on infectious diseases, vaccine delivery systems, and antiviral strategies, with notable contributions to SARS-CoV-2 diagnostics and therapeutics. Please help me welcome Professor Lee.
Thank you for the introduction. Good afternoon, everyone. Welcome to the Session 3, Emerging Vaccine Platforms from Concept to Reality. I'm honored to serve as the chair for this session. Today, we will explore some of the most innovative vaccine technology being developed around the world, from self-amplifying mRNA platform to microneedle array system and advanced lipid formulation. These cutting-edge approaches are shaping the future of global vaccine preparedness and response. We are joined by 3 distinguished speakers who each bring a unique perspective and expertise in advancing novel vaccine platform. Without further delay, let's begin our session. Our first speaker is Professor Gerald McNally, a leading expert in molecular biology at Karolinska Institute in Sweden. He has more than 2 decades of research experience focused on alpha virus infection and self-amplifying mRNA vaccines. Today, he will share his insight into the application of self-amplifying mRNA technology in combating pandemic and other disease. Dr. McNeely, the floor is yours.
Okay. Thank you very much, and thank you to the organizers for this very kind invitation to come and present. So, indeed, I'm the professor of molecular virology at our department at Karolinska Institutet, and I will tell you a little bit today about our platform for developing self-amplifying mRNA vaccines. How do I... Okay, so we've heard a little bit today about self-amplifying vaccines. I was very happy to see it mentioned in a number of talks. They are simply based on replication-competent mRNA molecules, which are derived from positive strand RNA genomes, typically alpha viruses, but some others have been described. And perhaps this is an oversimplification, but I like to think of them that they combine the best of the old-school live attenuated vaccines that induce very potent immune responses against particular vaccines, but against particular diseases, but are very labor-intensive to produce. And the new world of mRNA vaccines, which are very versatile and can be rapidly adapted. And the platform I'll tell you about has been in development at our department for, well, about 30 years. The Semliki forest-based RNA system, we've only recently, of course, started calling it the self-amplifying RNA system, but we called it the replicon system or the SAV-based system. So this is the virus. I want to tell you a little bit about the biology of the virus, because one of the points I want to drive home is that the biology of these viruses is a little bit more complex than perhaps people think when they draw these self-amplifying vectors on their slides. So we want to talk a little bit about the biology. So Semliki forest virus is one of the prototypic alpha viruses, and it's frequently used model viruses and has been used as a self-amplifying vaccine platform because it has very, very wide host range and very fast replication, very high expression of viral proteins. And it's a positive strand RNA virus, which means that upon entry into cells, it's translated, its genome is an mRNA. And we have 2 open reading frames that are important in this virus. One is in the light green here, the non-structural polyprotein, which is the proteins that come together to form the polymerase of the virus and also to interact with the host cell. And then in the dark green, we have this structural protein, and this is where the antigen is placed in the self-amplifying vectors. And what you see on the right side of the slide is just looking at protein expression and viral replication. Within 3 or 4 hours after infection of cells with this virus, the cells really are just factories for producing the protein, the coding regions that are held on this 26S mRNA. So a very, very efficient production of those proteins. And the viruses grow to very, very high titers. So looking at the alpha virus life cycle, this is from a review I published recently with some colleagues. The virus enters by receptor-mediated endocytosis, and the genomic RNA is released. And as I told you, it's first thing that happens, it is translated, and we get a large polyprotein, which is all 4 of these non-structural proteins produced together. And then there's a temporal control of the processing of this polyprotein such that the P123 and fully mature NSP4 is the polymerase for the producing new negative strand templates from the incoming positive strand. And then the fully processed NSPs 1, 2, 3, and 4 form the polymerase for making new positive strands, and that's shown here in the center. So the negative strand RNA serves as the template for more production of more genomic RNAs, which are then fed back into translation, amplifying the system, making more negative strands. And then later, late in Semliki is 3 or 4 hours after infection, but at later times, the cell's really just producing. The only mRNA that's being produced in the cell is this subgenomic RNA, which contains the reading frames for the structural proteins. And the structural proteins expressed at very high levels and go to form the nucleocapsid, and then the spike proteins are co-translationally translocated in the ER and expressed on the surface for assembly of new virions. So back in the late 1980s and early 1990s, my colleague Peter Liljeström cloned the genome of Semliki forest virus, and it was not as trivial as it is these days, but the first thing that Peter and his team did was to remove the structural protein genes and replace it with, in this case, a reporter gene, beta-galactosidase. And this is basically this is the simple part of the presentation, where we say that we can make these self-amplifying RNA molecules expressing any antigen of choice in that position. And you might ask why I'm talking about this. Is the work of Peter Lillestrøm over many years, but just want to mention that I was a postdoc with Peter, and I was recruited as a virologist with the idea that we would by learning more about the viruses, we would be able to make the replicon system better by introducing mutations to change the biology of the virus. And I went on to form my own group independently of Peter, and now that Peter is retired, we decided to combine our groups so that the replicon system is now in development again now in this molecular virology group that I head of. So if we look back at the life cycle of the virus, what's left behind when we introduce this molecule, and we can introduce that in a number of ways that I'll tell on the subsequent slides. But again, we have this production of the replicase, exactly like I mentioned earlier, and this very efficient expression of the subgenomic mRNA, but now you have high-level expression of antigen instead of the structural proteins. And if the antigen is a glycoprotein, it will be translocated into the ER and expressed at very high levels on the surface. And importantly, we put some work into over the years in understanding the signals that are given off by infected cells. So there are, well, we tend to refer to them as adjuvant signals, because these cells that are harboring these replicons are giving off signals to the immune system that would then lead to the very strong immune response that these viruses generate. And of course, that's a key part of this system as well. And so learning more about how the virus controls those signals will allow us to make better saRNA replicons in the future. So how do we get the RNAs into the cells? There has been a number of methods developed in Peter's lab over the years, the first being what we call VREP, which is production of virus-like particles, where we package the RNAs in trans using co-expression of structural proteins. We also then more recently, well, at the very start and more recently have been developing saRNA replicons, or rRep. We also have a version of the platform where we can introduce a DNA plasmid, which has a CMV-driven cassette, which will then launch the production of an saRNA molecule, but allowing it to be delivered as a DNA plasmid, DNA vaccine. And then another system that we worked on for some time but stopped was a so-called Trojan horse, or a TREP, which contains a double subgenomic promoter. So you have expression of the antigen and the viral structural protein. So you would have actually an infection in the animal or in the patient. But we haven't been working more on that. Obviously, some safety issues to consider there. But first, looking at the VREP, so this was the system that was used most over the years in Peter's lab. And this is simply by co-transfecting cells with your rRep, your replicon, together with two truncated replicons, where the NSP region has been truncated, but containing the genes for the capsid and the spike on two separate helper molecules. And when you introduce those three RNAs together, the NSP polymerase from the rRep will replicate itself, but it will also replicate in trans the helpers. So that therefore you would get high-level expression of the capsid and spikes, and then you would have packaging. And these particles down here on the bottom of the slide would then be indistinguishable physically from Semmelweis virus particles, but they would contain the replicon RNA rather than the viral genome. And this was a very useful technology. Over the years, we made many stocks. Our freezers are full. And they were very useful for small and medium-scale immunization experiments, but it was very obvious at the time and now in retrospect that this was never going to be scalable for larger vaccine studies or as a product. And so looking back, we now start working much more on the rRep and DREP iterations of the platform. And so the use of SFV vectors as saRNA replicons actually dates back to 1994. So this was one of the very first papers on RNA vaccinology, and this is where Peter's group were comparing immune responses to influenza virus NP in mice immunized with either the VREP, this is the modern technology, the VREP, or the rRep. And this was just RNA taken directly from the transcription reaction and injected into the muscles of the mice, completely naked, no LNPs at all. And surprisingly, I think at the time, they did get a very strong antibody response, and it was boostable by a second immunization, but was never able to compete with the introduction of the RNA using a virus-like particle. And for that reason, this technology wasn't worked on very much more Although there was a paper later where the team took 3 different RREPs, and again, just immunization of mice with naked RNA, it was able to provide protective responses against a tick-borne encephalitis virus, LIV, also influenza virus, HA, and respiratory syncytial virus. But those were small studies just using naked RNA. Much more recently, and I was happy to see this project mentioned on an earlier talk this morning, we have now, together with Dokeun Kim and Man-Ki Song here at IVI, we've been developing RREP expressing different forms of the MERS-CoV spike protein. And we have these 4 versions here. This is now just the antigen that's depicted. And we're able to go back to my great pleasure to some of the work that I had done back when I was in Peter's lab, where we developed an enhancer, a small region of the viral genome that's important for efficient translation. And when we put that into the replicons, and we were wondering whether we could get higher expression of the gene. And in this very simple experiment here, we did a Western blot just to look at the antigens that are expressed. And indeed, the 2 replicons that contain this enhancer are producing much more protein as detected by serum from convalescent patients. So we're very excited about doing immunogenicity studies here in Seoul now with these replicons, which I hope will start very soon. I know we're going to discuss it more tomorrow. And then just at the end of the talk, I just want to tell you also about this DNA-launched vector. So that's very simple technology where you just exchange the SP6 promoter for in vitro transcription with a CMV promoter and also some termination signals at the end, the 3' end. And Peter's lab were working on this for many years. And at the start of the COVID pandemic, we decided to join forces. This is when we decided to combine our labs. And we made 2 DREP constructs expressing either a cell-associated or secreted forms of the founder strain of the SARS-CoV-2. And this is a very, very small mouse immunogenicity study, but we were happy to see that 4 out of 5 mice gave us measurable neutralization responses after 1 dose of this DNA plasmid, and which was then boostable with DNA or with protein. And then just to finish off on a phase 1 clinical trial, which is ongoing, and we're going to see the results very soon. This is set up by Peter's lab before we joined, but a DREP construct expressing GP140 from HIV is now undergoing a phase 1 clinical trial for safety and efficacy, and that DNA has been introduced either alone or in combination with clade-C envelope protein in healthy HIV uninfected adults. And it's a number of doses and delivered by this PharmaJet needle-free system. So we look forward to seeing the safety and data from that very soon. So with that, I'll wrap up, and just to say that, we have this platform now that can be, where the mRNAs, self-amplifying mRNAs, vaccines can be introduced in different ways. And I just want to underline the point that we do believe and we know that the biology of these alpha viruses is much more complex than we think. We know that there are a lot going on. We know that there's things happening in the cells that we don't know of, and so I think there's an opportunity now and in the future to tweak these, to learn more about the viruses in order to be able to tweak the saRNA systems to make better replicons, better vaccines. And so this is my group as it is right now, with Peter associated. And we have a number of funding agencies, the majority of which are funding our virology projects to learn more about the biology of chikungunya and Ross River and Semliki forest virus, with the exception of IVI, which are very generously supporting the development of the MERS RREP platform. So I'll stop there, and thank you very much for your attention, and thank you again to the organizers for this invitation.
Thank you very much, Charles. Thank you for your fascinating presentation on self-amplifying mRNA vaccine. Let us now move on to our next speaker. Next, we have Dr. He Jin-ho, Research Director at Quad Medicine and pioneer in microneedle vaccine technology. With your unique background bringing bioinformatics and diagnostics, Dr. He will introduce us to the latest advancement in microneedle array patch technology and their potential to revolutionize vaccine delivery. Now let's hear from Dr. He.
Thank you for nice introduction. I'm very honored to have a chance to introduce a microneedle array patch, which is a novel vaccination technology. Microneedle array patch is a small patch consists of hundreds of micron-sized needles which can penetrate the outer layer of the skin. It is less painful. If we measure the VAS scale, it is under 1, like 0.5 pain score. And it is easily applied to the skin and self-administered. That means we don't need a doctor to apply a vaccine. And it delivered large molecules or hydrophilic compounds. And finally, and very importantly, it is thermostable. We don't need a cold chain. So we can save a lot of logistic cost. This slide is very interesting to me because we think about the target penetration layer of microneedles. Microneedles can be designed to target the epidermis or dermis based on their application. If we think about the conventional hypodermic needle, it goes to the hypodermis or muscle layer based on the length. But microneedles can be targeted to epidermis or dermis. And transdermal patch goes to epidermis layer. Then, if we think about the immune cell density across skin layers, which is mentioned in the morning plenary section, immune cells like APCs are most abundant in epidermis or dermis. And the muscle layer, they are least abundant. Even though they are least abundant, most vaccination is administered via intramuscular injection. That means in case we target epidermis or dermis, it could be advantageous. But we don't know how effective are they. That's why when we developed microneedle patch, we hypothesized that if we target epidermis and dermis, it could be as effective as IM injection, or it could be better. To address this hypothesis, we generated different kind of microneedle array patch and tested against different vaccines. Conventional microneedle patches major categories are 5 types: solid, coated, dissolved, hollow, and separable. You may be familiar with all these types of microneedle patches. And it follows their own pathways. Let me walk you through the core platform technologies. Coated map. Coated map is a very simple approach. A biopolymer is coated with APIs. And when penetrated into the skin, the APIs start to dissolve and penetrate into the skin. As you can see from the figure, CMAP can be applied like we use a bandage. SMAP is our company's unique technology. To apply SMAP, we need an applicator. When SMAP is applied to the skin using an applicator, it's separated in the middle according to the separation line. And the tip of the map is inserted into the skin, and the microneedle tip has APIs. And APIs start to melt and penetrate into the skin. Next one is powder attached map. To make powder attached map, first we need to make a powder form APIs. To make a powder form, we lyophilize liquid drug substance and then milled. And it turned to powder, API powder. And this powdery form is coated onto the biopolymers and sticky layer coating. Next, powder is coated like a corn dog, sugar powder over the corn dog. And powdered APIs are firmly attached to an adhesive layer of biopolymer map. Next, map penetrates the stratum corneum and powdered API are located in the intradermal layer. Finally, after 5-minute administration, map is removed and the APIs are rapidly absorbed into the skin. Each map has unique properties, and I think there is some match between specific map platform and specific vaccine. I will show many examples, and you will understand what does it mean. These are the latest updates in our platform research. First, I brought 2 examples of coated map, which is applied to hepatitis B and pentavalent vaccine. If you look at the upper layer, 2 figures, blue dotted box is intramuscular injection, and red box, dotted box is microneedle array patch immune responses, and with different formulations. As you can see from left and right, log G titer or log G1 and log G2A titers, which is humoral and cellular immune response, which the immune responses were as comparable as IM injection. For pentavalent vaccine, instead of mixing all 5 different vaccines together, we just make one map for each vaccine and make mosaic type vaccine applicator. The 5 figures box plots are the immune responses of each map. This elicited antibody titers comparable to those achieved by IM injection, and we didn't use any adjuvant here. Next example, separable map example. One is H3N2K9 influenza vaccine map. The other one is human papillomavirus map. As you can see here, MN, microneedle set, is comparable to IM injection, whether we use HI assay or SN, serum neutralization assay also. For human papillomavirus vaccine map, it delivered into the buccal mucosa using map-induced anti-HPV IgG titers similar to those induced by IM injection. This figure is a serum IgG level, and the red circle is a commercial HPV vaccine, and red square is a human papillomavirus map. You will see the performance here. The third one is a PMAP application. For this, we collaborate with Moderna. So we used the Moderna mRNA LNP vaccine, PMAP, and it induced IgG in sera, bottom left part figure, and we tested neutralizing antibody titers. These 2 figures show that the first set is IM, next one is ID injection, third one is MOC, and 4 to 6 are PMAP 1.25 microgram dose, 2.5 microgram dose, and 5 microgram dose. So especially at 5 microgram dose, the titer value were comparable with IM or ID. We also checked bar fluid, long washy fluid, IgG titers. Also, at 5 microgram PMAP group, it shows the similar response, immune response to IM or ID. This example is Shigella vaccine PMAP. To test this Shigella vaccine with the PMAP platform, we collaborate with the GSK, and we obtained the Shigella vaccine from them, and we checked humoral immune response and long-term immunogenicity. But this time, we also tested the PMAP and CMAP, both PMAP and CMAP. At 5 microgram dose, the IgG titer value were similar to IM. CMAP, PMAP showed a similar performance. We also observed the serum bactericidal activity titer, also similar result obtained. We checked long-term immunogenicity induced by P and CMAP. When we measured the total long-lived plasma cell percent in bone marrow cells, IM, CMAP, PMAP, even PMAPs are comparable or slightly better result. To measure long-term immunogenicity, at first we measured till 6-week WPI, but for long-term immunogenicity, we lived it till 21 weeks post-immunization. And as you can compare from first to the fourth figures, the immune response did not reduce. It maintained almost the same. And the performance was also similar. IMC and PMAP were similar. The final example is measles vaccine PMAP. This slide, I think the measles vaccine and the PMAP is a perfect match because when we observed the PR and T test result, it was very exciting result because the first one is Merck and the second one is commercial human dose vaccine. And I'll only explain the human dose only. And the fourth one is vaccine, liquid form vaccine, human dose. And liquid form vaccines are administered via SC injection because usually measles vaccine are administered subcutaneously. So these liquid form are subcutaneously injected. And the PMAP is applied just the way we always do. And the PRNT result concentration showed dramatic increase, means when we consider the seroprotective threshold is 120, which is the dotted line in the bottom, then it is way over the seroprotective level. It shows a strong immune response compared to even liquid form. That's why we first couldn't believe this fact and tested again. And second time, we also the concordant result with this. So that's why maybe specific vaccine have a matched platform, MAP platform. And here, measles vaccine is a perfect match with the PMAP. So from these examples, we have a confidence that MAP platform is as efficient as intramuscular injection. To wrap up, I'll briefly introduce our company. Our company specializes in developing mAb platforms for vaccines and drugs. We have in-house capability and develop mAb production equipment, and we use GMP-compliant facilities to produce mAb products. And we are collaborating with many pharmaceutical companies and vaccine organizations for R&D. These are business partnerships. We are collaborating with many leading pharmaceutical companies to develop mAb products for existing licensed drugs or vaccines using our innovative drug delivery system, including LG Chem, GSK, Hanlim, Kwangdong, and Ilyang Pharmaceuticals. Thank you for your attention.
Thank you, Dr. He, for the enlightening overview of microRNA technology and the clinical potential. Our final speaker is Professor Hyuk-Jin Lee from Seoul National University, an expert in non-viral gene delivery systems. He will present innovative approaches using ionizable lipid nanoparticles to enhance mRNA vaccine efficacy and safety, highlighting the role of engineered lipid system in next-generation immunotherapy. Dr. Lee, please begin.
Okay. Thank you for the nice introduction. It's my pleasure to be here. So I have an engineering background developing non-viral gene delivery systems. And today I'm going to go over some recent progresses of my laboratories developing ionizable lipid for mRNA vaccines. So as you seen in this slide for non-viral gene delivery system, there are two exact roles of these delivery systems. First one is to protect genetic payloads such as plasmid DNA, messenger RNA, and synthetic oligonucleotides. The second one is to facilitate the intracellular delivery of these RNA payloads to the inside of the cells. Mostly, we are very interested in utilizing messenger RNA for expressing various proteins and using mRNA-based approach. So for lipid nanoparticles, they are composed of 4 different lipid components, and payloads we usually focuses on nucleic acid-based drugs. And as you can see here, ionizable lipid actually has important roles. And when we formulate these particles, all these 4 different components have functions. And I like to actually emphasize the roles of ionizable lipid here. So when we actually synthesize ionizable lipid, the bottom part, and when you look at a chemical structure, the majority of the ionizable lipid has tertiary amines that can actually further protonated at acidic conditions. And this cationic character change actually initiates the charge-to-charge interactions with the anionic drugs such as messenger RNA. So we actually formulate lipid nanoparticles at low pH conditions, facilitating the electrostatic interaction between ionizable lipid and RNA payloads. Once they are formulated, we actually do a buffer change to neutral, and this actually taking care of the excessive charge of the lipid formulations. Once they are administered to the body, what happens is that the surface of lipid nanoparticle is actually exposed, and a lot of seroproteins get absorbed on the surface. And in typical lipid nanoparticle formulations, ApoE usually absorb on the surface, and during a circulation, the cells that expressed LDL receptors recognize these particles and allows the intracellular delivery of these LNPs. Once they are internalized, these particles actually get entrapped in the anosome, and the anosomal pH is actually acidic. And when acidic conditions happens again, those ionized lipids become charged again and try to interact with the endosomal membrane. And this actually initiates the endosomal leakage such that we can actually deliver RNA payload to the cells. So these are some commercialized ionized lipid. Above, there are 2 ionized lipid for vaccine applications, such as ALC0315 from Acuitas and SM102 from Moderna. And when you look at down left, there are ionized lipid called MC3. This one is actually first FDA approved ionized lipid for the delivery of a synthetic oligonucleotide called siRNA for treating polyneuropathy. And the bottom right is one that we developed in my laboratory and has been commercialized by SD Pharm. When you look at the different structures of ionized lipid, we can actually tell how many of the tertiary amines that can be existing in one lipid structure. As you can see here, Moderna's and Acuitas ionized lipid has a one functional tertiary amines that can be charged. However, in my laboratory, we use a bulkier amines that can actually charge more than one. So utilizing more tertiary amines means that we can actually reduce the amount of the ionized lipid in our formulation. So when you look at tables of formulation parameters, a conventional ionized lipid, you usually require use of ionized lipid, at least 40 to 50% in order to initiate endosomal release. However, the ones that we made, we utilize less than 30% of ionized lipid in our formulation. Then, you probably ask, how do you actually develop or identify these ionized lipid? In my laboratory, we actually do a synthesis called combinatorial synthesis. So we screen ionized lipid by making huge libraries of this compound by reacting ionized lipid head, linker, and tail. And below, I actually show 4 different ionized lipid that can be used for the different applications. So once you identify, proper ionized lipid, we utilize a microfluidic mixing system to generate lipid particles. And when we formulate this particle, we measure the size, also the distribution of the size. Also, the important factor is encapsulation efficiency. And also, we evaluate charge reversion at certain pH. And today, I'd like to share one of the studies that we did for the vaccine development. This work was collaborated with KDCA. First, we try to identify novel ionized lipid that can induce immune triggering effect once they are administered as vaccines. So we first started with amine heads called N4, and we evaluated different tail structure to make ionized lipid. And as you probably know, many of the vaccines are now administered in IM injection. So we actually tested the mRNA vaccine administration by IM and evaluated the expression profile with the reporters. After that, we identified one novel ionized lipid called N4Z, and we evaluated further for the preclinical studies. So we formulated this particle and encapsulated human erythropoietin encoding messenger RNA and did IV infusion for this. And we evaluated how good they are to deliver those mRNA cargo to the cells. And we confirmed that those N4Z actually show very good expression profile as comparable to the Moderna's formulation. After that, we evaluated the biodistribution by incorporating copper, radio-labeled copper to our LNP. And typically, LNP circulates and they accumulate in the liver. And over time, these LNPs can be removed from the body. And these are the toxicity profile of identified LNPs in mice. So we try a single dose, also the repeated dose. And as you can see here, by comparing with the Moderna's formulation, we actually saw comparable toxicity profiles. And the other one, we actually did NHP studies with this formulation. We first tried to figure it out whether the repeat dose actually produce the same amount of the proteins in the NHP models, because many of our formulation using a PEG can actually cause the production of anti-PEG antibodies. So after the first injection, and in many cases, the protein production yield actually diminishes. And nowadays, people are thinking about administering those vaccines or therapeutic agents more than one time. So we want to make sure repeated dosing can produce a reproducible expression profiles. After that, we also evaluated blood markers like AST and ALT and others, and those markers seem to be very comparable. And one other study that we performed with my laboratory, we wanted to actually see the adjuvancy efficacy of the lipid formulations. So by injecting those formulated LNP, IM, we try to figure it out, how LNPs can show the adjuvancy effect. And we did single-cell RNA sequencing, and depending on the ionized lipid that you use, we found that the delivery to the specific cells can be different. So when you look at the bottom left, the Moderna's formulation, once they are administered into the muscles, the mRNA actually goes to the macrophage. Also, the expression of those mRNA is high in macrophage, which is a very distinct phenomena. Because we tested a different ionized lipid, and we saw this phenomenon was a very unique. And as I already mentioned, because once they are administered in IM, those LNPs with a different ionized lipid tend to go to the different types of cells. And we actually evaluated whether the chemokines and cytokine release profile might be different depending on ionized lipid. And as you can see here, macrophage cells, Moderna's formulation actually show very good chemo and cytokine release. And the other tested materials actually show those releases based on fibroblast. And we also evaluated early chemokines and cytokine markers like MCP1 and IL-6 upon IM injections. And we found that Moderna's formulation actually show very strong MCP1 release, also the IL-6 inductions. And because we try to figure it out, the proper ionized lipid for the vaccine applications, and one that we identified is called M4C, inducing proper chemo and cytokine release upon IM injections. And we evaluated that total antibody productions, also the also the body weight change, and also evaluated survivals. And we found that M4Z actually show very good productions. And then this is last part, almost the last part of my talk. So we found that those branched lipid structure actually required for the proper stimulation of immune systems. And then we are now excited to actually change the head structure to find out ability to target different types of cells. And in my laboratory, we found one of the candidates that actually facilitates the delivery of those mRNA vaccine to the macrophage cells. And we tested first in cell lines, and after we did that, tested with the PBMC. And this is quite interesting because the targeting to the macrophage cells and immune cells can be actually utilized for further cancer vaccine applications. So once this formulation is injected it by IV administration, these particles tend to go to the spleen. And this is a quite a different profile that we usually see with conventional LNPs, because LNPs usually go to the liver because of the LDL receptor-mediated uptake. And we actually evaluated that in a spleen site, what cells actually get this mRNA LNP, and we found that majority are actually macrophage cells. So we're trying to use this novel ionized lipid as well as LNP to use as cancer vaccine applications. Conclusions, so immune stimulation ability is actually very important for mRNA vaccine applications. So finding out proper ionized lipid is very necessary. And for proper cellular response, we believe that delivery to the macrophage and monocyte is also very important. Thank you for your attention, and these are some acknowledgement slides. Thank you.
Thank you, Hyuk-jin, sharing your innovative work on ionizable lipid for mRNA delivery. We now have 10 minutes for questions for the audience. Please feel free to raise your hand to ask a question. Let's open the discussion.
Hello, thank you. I'm Shin-hye working with Doctors Without Borders. My question is for Director Hae-jin Hu from Quad Medicine. Yes, so I saw with great interest your presentation about the measles patch vaccine. Yeah, I'll wait until you get to the stage.
Okay.
Yeah.
I'm ready.
Okay, so I have three actually quite simple questions related to that technology. So first one is what is the timeline for commercialization, and what are your plans for the regulatory pathway? Where do you seek to? What is your timeline for the regulatory pathway, especially for the global market? And when you were trialing it, how long did the patch have to be in contact with the skin to show the level of efficacy? So yeah, those are my three simple questions.
Would you repeat the first question? Yeah.
The commercialization timeline.
Commercialization. That is the most commonly coming questions. Actually, we need to follow the regularization first. So we are Hep B vaccine. We are pursuing the phase 1 clinical trial. And for others, we are applying IND. So we need to take time. And it is not just our problem. All mAb platform companies are pursuing their clinical trial 1 or 2. And I think this technology is just entering the clinical phase right now. So I hope within a couple of years, any company, any mAb company can produce commercial products. And then other companies also will be, easily follow the tracks, I hope. Yeah, and that is the first and second question, right? And yes. The third one is, I'm kind of nervous, so that's why I cannot remember all the questions. So can you repeat the
third question was how long the patch had to be in contact with
that
the
part.
skin.
Yeah, it depends on the mAb platform. For SMAP, we use applicator. So it is like a ballpoint pen. Right after applying the mAb, then it just breaks. So we don't need to wear. The tip is just inserted into our skin and it dissolves slowly. So we don't need a wear time, but CMAP or PMAP, for CMAP, currently about 10 minutes we need to wear, at least 10 minutes. For PMAP, as I already presented there, it takes 5 minutes. So after 5 minutes, we can just take it off. Yeah.
I have a question to the Professor Hyuk-Jin Lee. It was the kind of the dramatic targeting by the lipid, right?
Okay.
So actually, I learned from you the LNP can make the protein corona and then target it. So my question is, it worked very well in mouse. Did you check it work same way?
So
in
basically,
human?
when we changed the formulation parameters as well as ionized lipid, that actually changed the surface chemistry of this lipid nanoparticle, and basically we used endogenous protein to direct those particles to the different tissue or cells. And as you already mentioned, many of the early studies actually they cannot be reproduced in a higher animals, but for spleen targeting LNPs, we try to actually do our studies in a NHP setup. And some of our initial studies actually show that that can be actually deliverable to the spleen.
Oh, okay, thank you.
Yep.
I have a question to Professor McNally. Thank you for the great talk. And I have a question concerning the fidelity of the replicase. Since it uses viral replicase, and as far as I know, it doesn't have a proofreading function. So in that scenario, would that cause raise a mutation and cause ultimately the neoantigen due to the low fidelity? And if so, then the fidelity can change, can be variable by the mRNA length and context. It is context dependent. So is that being considered during the mRNA design?
Yeah, well, you're absolutely right. There's no proofreading in this, but with the replicons, there's also no replication. So there's no transmission of the replicated product to the next cell. It's always we always go back to the DNA plasmids to transcribe. So the error rate would, in effect, be very, very low. So you'd only have one in every, I don't even know what the numbers are, but maybe there would be some mutations in the antigen, but I would expect them to have very little effect, except maybe in the T-rep platform, which I didn't talk about, which is then transmitted. So there, indeed, you would have a problem. But there, I think the problem would be more in terms of recombination rather than point mutations. But at least in the saRNA delivered as RNA or even as DNA replicons, there's the danger of having the low proofreading, I think, is minimal. Thank you. Thank you.
Yes.
My name is Haesuk Jung from the KDCA. Thank you for the great presentation. I have a question for Dr. McNally. The immune effect of the DRAP system was impressive. What kind of delivery method of the used? Was the electroporation or something like LNP?
The DNA, they were done by electroporation, yeah. Injection followed by electroporation. We've never used any LNPs, actually. So the study we're going to do here with IVI will be the first time, to my knowledge, that the Semliki replicons have been used in combination with LNP. So we're very excited about that. The DNA was electroporation. Yeah.
No further question.
Sorry, I forgot to mention in my presentation. For measles vaccine study, we collaborated with the IVI. Thank you.
Thank you once again, all our speakers, and to each of you for your attention. Enjoy the rest of the Global Vaccine Forum. Thank you.
Thank you, Session 3 Chair and speakers. We'll now begin the fourth and final session of today's forum, Korean biotech's leadership, developing and manufacturing next-generation vaccines. Dr. Hani Kim, Executive Director of the Wright Foundation, will chair this session. Dr. Kim's educational background began in biology, later extending into public health. Her career includes positions as a postdoctoral researcher at the Max Planck Institute of Immunobiology, followed by roles as a researcher and professor at Toronto General Hospital and the University of Toronto. She also served as a research director at Johns Hopkins School of Public Health and worked as a program officer at the Gates Foundation. She has published articles in Translational Medical Research and the Political Origins of Health Inequity. Please join me in welcoming Dr. Hani Kim.
I would like to thank you all for staying in the session for this very last session before you head to the cocktail reception. And let me assure you, you will not regret staying until the very end, because now we are going to switch gears a little bit to hear from Korea's three leading vaccine manufacturers to better understand what they're currently pursuing for next generation vaccines. So first, I'm going to invite our first speaker, Professor Jae-hwan Nam, who is also the CEO of SML Biopharm. Dr. Nam is a professor at the Catholic University of Korea, and he also founded SML Biopharm in 2021. His company, and we are going to hear more about its activities, is focused on developing various medicines using mRNA platforms, which we've heard a lot about today. And he's already contributed immensely to developing hepatitis C virus models when he was training as a postdoctoral scientist at the NIH. So, Professor Nam, please, the floor is yours.
Thank you very much for a nice introduction. Today, I'm going to talk about the bridging innovation model between SML Biopharm as a startup and Eubiologics as a big company to develop this COVID-19 mRNA vaccine in Korea. Today, I will break this topic into the two parts. In the first part, I will just simply mention about the general aspect of the mRNA vaccine and unique characteristic of our mRNA technology. In the second part, I just simply talk about why the SML Biopharm can collaborate with Eubiologics to develop the mRNA vaccine in Korea. Okay, let's move to an mRNA vaccine 101. As a previously speaker already talked about that, but I just simply mention about that. As everybody knows that mRNA vaccine require two essential components. One is mRNA expression platform, the other is delivery tools. There is three different mRNA expression platforms, linear form, circular, and self-amplifying mRNA platform. These three different mRNA expression platforms has a different characteristic. Previous speaker already very well mentioned about that. Anyway, if you can choose one of them dependent on your purpose. So, however, the most common the mRNA expression platform is a linear form. So, in this talk, I just focusing on the linear expression platform. And the other is there are several delivery tools. The most common delivery tool is a lipid nanoparticle. Dr. Lee also present about that. This lipid nanoparticle has some four different lipids, phospholipid, ionizable lipid, and cholesterol and piezo lipid. However, the key challenge step is manufacturing process. I am working in the academic part, so before I founded my company, I don't know the importance about the manufacturing process. So manufacturing is a very key challenge step. Anyway, as you can see, the mRNA manufacturing process is based on the chemistry. So this is a very important characteristic. So, okay, there is a two-way in which mRNA vaccine can function in our body.
First is mRNA vaccine expressed the protein can stimulate some specific immune cell, T cell and B cell, through the antigen presenting cell, especially the dendritic cell, and allowing for the activating the acquired immune responses to get some good neutralizing antibody and some specific CD8 T cell for the cytotoxic T lymphocyte response. The well-known example is Moderna COVID-19 mRNA vaccine. And the other is we can apply this system to develop the therapeutic mRNA vaccine. The only difference between preventive mRNA vaccine and therapeutic mRNA vaccine is expressed the protein. So the preventive mRNA vaccine usually expresses the pathogen protein. In contrast, a therapeutic mRNA vaccine expressed usually the tumor-associated antigen or a tumor-specific antigen as a neoantigen. So you have to remember that the only difference is expressed protein. The other is just very similar. The second way is mRNA vaccine expressed the protein can restore biological function. I mean, mRNA vaccine expressed the protein itself can be effective in our body. The example is Moderna developed the IL-2 fusion protein to help treat the autoimmune disease. Also, we can express the therapeutic protein or some specific human monoclonal antibody using by the mRNA expression platform. So the mRNA drug has a very broad application, including the preventive mRNA vaccine for infectious disease and some therapeutic mRNA vaccine for the cancer. We can express some specific therapeutic protein or some specific human monoclonal antibody. We also expressed the Cas9 system for the genome editing, and even though we can express some specific gene in the stem cell for the cell therapy. Okay, let's talk about the specific characteristic of our mRNA technology. Our mRNA expression platform has a very specific 5' UTR and 3' UTR sequences. Especially our expression platform has a circular poly-A tail structure. This circular poly-A tail structure showed some resistance of RNase degradation. And so our circular poly-A mRNA platform clearly showed higher expression level compared to Pfizer and Moderna-like structure in the several cell line. The NO10 is muscle cell line, and HEP2 is the liver cell line. Also, our circular poly-A tail structure clearly showed some higher level expression and longer stability in the firefly Rush project and the human EPO in the mouse body. So our mRNA expression platform showed some better than the others or similar in vitro system and in vivo system. Okay, next is we used the artificial intelligence to design the lipid nanoparticle. We just focusing on the diamine core structure to design the ionizable lipid. Actually, we already published all data in the small in last year, so if you want to know some detailed information, you can read that paper. Okay, and next is we developed some novel lipid nanoparticle which have a different the lipid formulation. This novel lipid nanoparticle has a less toxicity and has a similar vaccine efficacy. Our SLP005 clearly showed some minimizing the reticulocyte decline and minimizing the ALT level increase after mRNA vaccine. So we believe this safe profile may be due to our SLP005 showed some spleen bias distribution, but we need more detailed research about that. Okay. This slide showed our present at the SML 1.0 and our future SML 2.0. You can see our ongoing project. We developed the COVID-19 mRNA vaccine with Eubiologics to produce large scale. We applied this system to some next pandemic pathogen, including the avian influenza virus. And we also developed the preventive and therapeutic mRNA vaccine against herpes simplex virus 2. And second is we developed this therapeutic mRNA vaccine against human papillomavirus induced the cervical cancer. And we applied this platform to the head and neck cancer and some combination therapy. We also developed some therapeutic mRNA vaccine against Epstein-Barr virus induced the cancer. Third is we developed the mRNA vaccine expressed the human monoclonal antibody against SFTSB. KCDC are supporting this project. So we applied this system to some other solid cancer and some chronic disease using by the mRNA expressed bispecific antibody. Finally, we developed the mRNA drug for the sarcopenia, and we are expanding this platform to the systemic sclerosis and some sarcopenia by GLP-1 treatment. So we are very actively looking for some good partners. Okay, let's move to the second part. The SMA Biopharm is a new biotech. I just founded in 2021, and we are a very small company. Our employee is only 13. However, we are very actively collaborated with many companies in this world. Eubiologics and Invent-Jurab is a Korean company, and Eubiologics is a French company, and Nano mRNA is a Japan company, and KTOG is a Swiss company. However, the Eubiologics is a big company, and then they have a very beautiful GMP facility to produce mRNA vaccine. So when KCDC announced the domestic mRNA vaccine project, we can find a good opportunity for the collaboration. As I already told you, SMA Biopharm have a very beautiful mRNA-based technology, including the expression platform and the delivery tool, lipid nanoparticle. Actually, we have our own patent. However, we don't have some manufacturing facility, but Eubiologics have a very nice GMP facility to produce mRNA vaccine. So by combining our innovative mRNA technology with the Eubiologics the facility, we can make a good success to develop the mRNA vaccine in Korea. Okay, the SMA Biopharm tried to move the therapeutic protein and some bispecific antibody expressed by mRNA platform from the preventive mRNA vaccine and the therapeutic mRNA vaccine. Thank you very much for your attention.
Thank you, Professor Nam. So we are going to get all the questions at the end. So I'm going to now introduce you our second speaker, Dr. Yeoncheol Shin, who is currently the head of development team at the GC Biopharma Corporation. He has been with GC Biopharma since 2016, so almost 10 years he's been leading this program, and he's played a key leadership role in developing various GC Pharma's vaccines. One of the most notable ones is he was the project leader in successful regulatory approval of GC seasonal influenza and varicella vaccines. And he is one of the principal investigators that are leading the mRNA vaccine awards that we've heard about that have been dispersed by Korean Disease Control Agency this morning. Dr. Shin, please go ahead.
Thank you for kindly introduction. Good afternoon. I'm Yeoncheol Shin from GC Biopharma, as just introduced. First of all, I'd like to express my sincere gratitude to the organizer for giving me the opportunity to speak at this valuable forum. Today, I will be presenting Jushi Biopharma's strategic approach to mRNA vaccine development. In particular, I will focus on our mRNA COVID-19 vaccine project, which is being developed with collaboration with the KDCA. Here is an overview of today's presentation. First, I will briefly share Jushi Biopharma's expertise with the mRNA LNP technology. Then, I will provide an update on the progress of our mRNA COVID vaccine development. Let me begin with our expertise in mRNA LNP technology. Our R&D division has successfully internalized both the manufacturing process and analytical methods across the full spectrum of mRNA LNP development. Since 2019, we have established dedicated teams specializing in mRNA and LNP technologies and have been actively engaged in a wide range of R&D projects. For mRNA technology, we collaborated with TuliLink to internalize both manufacturing and testing methodologies. For LNP technology, through a supply and technology transfer agreement with Acuitas, we directly acquired LNP composition, manufacturing process, and analytical methods. As many of you know, both TuliLink and Acuitas technology have been applied in approved mRNA COVID-19 vaccines. In addition, we have established a dedicated mRNA LNP manufacturing facility at our Hwasun plant. The suitability of this facility has already been verified through prior projects, ensuring that it is ready for production. Now, I'd like to turn to Jushi Biopharma's mRNA COVID vaccine project. Internally, we launched this program in mid-2024. As I mentioned earlier, we were able to accelerate the R&D timeline by leveraging the technologies and experience gained from our previous mRNA LNP project. I'll explain more in detail shortly, but our goal is to submit an IND application to the MFDS in the second half of this year. Our mRNA LNP formulation is built on a combination of our proprietary technologies and components that have already been clinically validated. In particular, for the cation repeat, one of the most critical components of LNPs, we established an in-house repeat library. From this library, we selected the most promising candidate based on performance and applied it so the LNP formulation used in this project. Using the selected antigen for our COVID-19 vaccine, we conducted a preliminary immunogenicity study in a mouse model. As you can see, we confirmed that our vaccine induces level of antibody, IgG antibodies, surrogate neutralizing antibodies, and CMI comparable to those of the licensed comparator vaccines. Next, I'd like to share the details of our ongoing non-clinical studies. Through consultation with the MFDS, we have confirmed the required non-clinical study components for the IND package. So we plan to conduct toxicity, pharmacokinetics, and efficacy studies in accordance with our 2025 timeline. This slide outlines our progress and plans for this year. Multiple activities are currently being carried out in parallel, including manufacturing, analytical method development, product characterization, stability testing, non-clinical studies, clinical planning, and IND documentation. And then this year's goal is to obtain IND approval within this year. This is my final slide. Through this project, our aim is not only to localize COVID-19 vaccine production, but also to clinically validate human safety of Jushi Biopharma's mRNA LNP platform. Once this is achieved, we believe it will enable us to enter clinical trials within 100 days in response to the next pandemic. As always, Jushi Biopharma remains committed to contributing to public health, both in Korea and globally. Thank you very much for your attention.
Thank you, Dr. Shin, especially for keeping to time. So moving right along to our last but not least speaker, Mr. Young-wook Park, who is the Senior Vice President and Head of Bio R&D at SK Bioscience. So he has been at SK Bioscience with over 20 years of experience in vaccine process development, as well as technology transfer and regulatory support across multiple diverse platforms. He led the development of Korea's first cell culture-based influenza vaccine. Some of you may already be familiar. It's called SkyCell Flu. Also, during the COVID-19 pandemic, Mr. Park served as a program lead for SK's recombinant protein vaccine, SkyCoV-1, that represents Korea's first COVID-19 vaccine. Mr. Park, please, the floor is yours.
Okay, thank you. Thank you for kind introduction. So, good afternoon. My name is Young-wook Park. I'm Head of Bio R&D of SK Bioscience. So I'm very honored to present here IVI Global Vaccine Forum today as your last part. So I'm going to introduce SK Bioscience cell culture-based influenza vaccine strategy from seasonal to pandemic preparedness. So here is the agenda for today. So I will briefly look at the influenza and cell culture influenza vaccine, and I'm going to share the SK Bioscience influenza vaccine program. So let me begin with influenza and cell culture influenza vaccine. So as you may know well, the post-COVID-19 influenza has re-emerged as a major threat with substantial disease burden. Although most influenza cases are mild and managed in outpatient care, a small but meaningful proportion progress to severe illness requiring ICU care, with a fatality about 0.1%. This may appear low compared to COVID-19 or other infectious disease, but due to the sheer volume of infection, influenza remains a serious disease. So this highlights that the need for effective and rapid vaccine platform, particularly to protect high-risk populations such as elderly. Vaccine mismatches remains a major challenge for influenza control. Vaccine effectiveness against H3 and 2 virus was less than other strains. For instance, when the vaccine strain matched circulating H3 and 2 virus, vaccine effectiveness was 33%, but dropped to 23% when mismatched. So this table showed the year-to-year variability in vaccine effectiveness. So it showed that the importance of using a vaccine platform that can reduce the risk of mismatch with circulating strains. During the 2023 and 24 season in Korea, the overall influenza vaccine effectiveness was modest, approximately 22.5%, and the protection was particularly reduced in adults aged 65 and older, emphasizing the need for more effective vaccine for the elderly population. Multiple studies reported that cell-based influenza vaccines are more effective than egg-based vaccine. These tables are data from a published study on the effectiveness of cell culture-based vaccine. And at SK Bioscience, we conducted the real passages test against H3N2 virus for the egg adaptation. So we observed 3 specific mutations in H3N2 antigenic site after 15 passages in egg. In contrast, cell-based propagation did not show such mutation, maintaining the original antigenic sequence. So these tables are data from a published study on the effectiveness of cell culture-based influenza vaccine. In pediatric and younger adult population, cell-based vaccine showed in fewer influenza-related medical encounters. These results show that cell-based vaccine works well in real-world conditions. So let's now take a look at SK Bioscience flu vaccine program. Sky cell flu is the first cell culture influenza vaccine developed and produced in Korea, approved in December 2014, almost 10 years ago. So it is a surface antigen vaccine with a favorable safety profile. Since 2015, SK Bioscience has supplied around 5 million doses of Sky cell flu annually, including about 2.5 million doses through Korea NIP. Sky cell flu is the only WHO pre-qualified cell-based influenza vaccine. It is currently exported to over 10 countries, including Southeast Asia and South America. SK Bioscience also contribute to the global influenza vaccine system by supporting WHO reference standard production. We provide cell-derived preliminary liquid standard for HA content assay via MHRA. So our material is included in the WHO standard material listing. It demonstrates that our active role in global flu vaccine system. Let's move on to the influenza vaccine for elderly. So as Korea and many countries enter an aging society, reduced vaccine effectiveness in older adults is becoming a major concern. Immunosenescence-related decline in vaccine effectiveness among older adults is well known, and thus enhanced influenza vaccines are recommended. The USACIP currently advises the adjuvanted or high-dose flu vaccine, such as Fluad or Fluzone high dose for senior aged 65 and older. There are 2 types of enhanced influenza vaccine, high dose and adjuvanted vaccine. So some studies suggest that adjuvanted vaccines can offer similar or even better protection compared to high dose vaccine. So these are the data taken from those studies. On the left, you can see that adjuvanted vaccine worked well, especially in older adults with underlying health condition. On the right side, the graph shows that adjuvanted vaccine triggered stronger polyfunctional T-cell responses than high-dose vaccine, which may help improve protection in the elderly population. So SK Bioscience is developing a differentiated adjuvanted influenza vaccine for the elderly based on our Sky cell flu platform. Our candidate has shown that comparable humoral and cellular responses to fluad, in particular in preclinical studies. And the process development and non-clinical tox studies are completed, with clinical trials scheduled to begin this year and regulatory approval targeted for 2029. And from here, I'd like to talk about H5N1 lastly. So as of January 2025, 964 human cases of H5 influenza and 466 deaths have been reported globally since 2003. Even though the risk to the general public is still low due to limited human-to-human transmission, but H5N1 remains a leading candidate. H5N1 is still considered a high priority pandemic threat. That's why it is important to prepare H5N1 vaccine in advance. In response to this, SK Bioscience also began developing H5N1 pandemic vaccine. Cell culture platform allows rapid manufacturing without depending on egg. SK Bioscience can supply 35 million doses, enough for 70% of Korean population within 6 months of a pandemic declaration. So our H5N1 grade 1 candidate has completed non-clinical studies using the same adjuvant as our elderly flu vaccines. And SK Bioscience was recently awarded a national project for rapid response avian influenza vaccine development, targeting the emerging clade 2344B strain. This is my slide for summary. So cell-based flu vaccine can offer improved effectiveness. Differentiated adjuvanted flu vaccine for the elderly can provide enhanced public health value. And cell-based manufacturing is critical platform for pandemic preparedness. And lastly, as a leader in cell culture flu vaccine, SK Bioscience is building a comprehensive flu vaccine portfolio from seasonal to pandemic vaccine, contributing to global disease prevention and health promotion. And thank you for your attention. Yep.
Thank you, Mr. Park. May I now ask all three presenters to come to the stage? I think we do have at least 10 minutes to answer any questions from the floor. Yes.
Oh, thank you for a very interesting talk. I have a question to Dr. Nam. I am an RNA scientist, so I was very fascinated with your circular protease structure, which looks very promising and very smart. But I'm wondering if I think mRNA molecule is such a very long and big molecule, so I think it would be very challenging to achieve the base pair with the terminal sequences and the 3' UTR. So I mean, it would be very challenging to achieve the intramolecular base pairing. But and so I have a concern of intermolecular base pairing of 2 mRNA molecules during the mass production. And of course, since mRNA is such a long molecule, there would be unwanted and non-specific base pairing within the mRNA. So I'm curious about whether there is any special secret or is it confidential? So, but anyway, it was very nice talk and thank you.
That is a very nice question. Actually, we don't, the RNA is a single stranded, so they can make some very many second structure. So we don't know the real RNA structure because our mRNA expression platform is the enriched 1 kb ranks, or when we inserted some gene of interest in our mRNA expression platform, the ranks of our mRNA expression platform is more than 5 kb. So we don't know the real RNA structure. However, we tried to divide some the sector, this RNA sector. So we make some at the end of our mRNA expression platform is less than 100 base or 200 bases. So we try to make a real structure using by that fragment of our mRNA expression platform. However, that is not real structure, so we don't know yet exactly.
Thank you. Can I ask one more question? Sure. Do you include heat denaturation and cooling after in vitro transcription to enable such a very wonderful base pairing? I mean, heat denaturation and cooling.
Denaturation?
Yeah, cooling for mRNA.
We don't try to do.
I see.
We don't know that. Yes.
I see. Thank you.
Thank you. Is there any other question from the floor? Yes, over there.
Yes, I would like to ask for the Dr. Park, SK Bioscience. Yes, I'm Sunbin Kim, and a research scientist in CleanLab of IVI. And I see the KCDA also contribute to the your bird flu project. So I'm wondering if there is any progress, even though it's very early stage. So could you please share your plan or current progress?
So actually that national project is recently awarded. So we are it's only just started from April. So actually currently we are importing the new recent circulating strain 23444B from CDC, and we are recently just starting the research working cell bank, research virus bank for the process development. So we are in very early stage now. So I hope I can have a chance later we share our process development result.
Any other questions from the floor? Yes.
Vaccine against H5 strain. I think using the subunit HA is much less immunogenic than other like H1 or something like that. So I think then they decide to use a whole virus inactivated vaccine. So what do you think about, your plan to developing vaccines? After you purify the whole virus from your cell-based system, you consider subunit or a whole inactivated vaccines for H5N1?
So most of all, our Sky Cell Flu system, we have already very efficient cell culture influenza vaccine manufacturing platform. So we try to develop H5N1 vaccine by using those existing cell culture-based influenza vaccine platform. So, and I think the key is adjuvant for the H5N1 vaccine. So as you see in my presentation, we screened many types of adjuvant to increase the humoral and cellular immunogenicity. So I think to summary, we will develop the inactivated subunit vaccine by our own cell culture technology with efficient adjuvant. Yeah.
Thank you. If I may use the chair's privilege, can I ask one question to Dr. Shin from GC Biopharma? Congratulations on having developed your own in-house mRNA LNP platform. Could you please comment on the thermal stability and safety profile that you are aiming for, or any preliminary data that you may have?
Yes, we already have many animal data for safety, and then we have a plan to submit IND application this year, and then next year we will perform clinical trial. At that time, we identify our mRNA LNP platform safety. Yeah.
Thank you. Please join me in thanking our last three speakers. Thank you so much.
Thank you, Session 4 Chair, Dr. Hani Kim, and speakers. At this time, I'd like to invite Dr. Jerome Kim back to the stage to deliver our concluding remarks to close the forum. Thank you.
Thanks, and just to let you all know that I'm standing or sitting between you and the reception, so I'll try to be brief, but there was a lot of information presented today, and maybe I'll start first. I don't know if many of you know that I was also a history major, but in 2010, Korea was informed by the US government that the vaccine it purchased in the US may be confiscated for US uses. That's national security law. Many countries invoked it during the pandemic. And at that point, Korea was 20% self-sufficient and the number 11 vaccine manufacturer in the world. I think that on the basis of what we saw today, that might be a little different. Actually, we know that Korea is now between 40 and 50% self-sufficient, and maybe on its way, maybe not to being number 5, which was one of the targets, but to being a larger global producer and provider of vaccines for public health. I think 20% of the vaccines in Gavi actually have some basis or manufacturer in Korea. So I think that's been very important, and we saw evidence of that today. So thanks to Eubiologics, the Korea Support Committee, and Seoul Cyber University as sponsors of this event and co-sponsors, and then, of course, to the KDCA, which was co-hosting this event. So thank you very much, Commissioner Ji. So I think we saw or heard many stories about mRNA. I mean, mRNA is definitely important and transformative technology. After decades of work, it was a technology that was proven during the pandemic, and it was very effective in preventing severe disease and death, with very high efficacy for those kinds of conditions, less certain for prevention of infection, which was of interest to many people. I think that one of the things that we saw today was that the platform is not as simple as just the mRNA, and that as we make incremental progress in the mRNAs themselves, the structures, the nucleotide composition, the untranslated regions, that we may start to see further incremental improvements in our ability to not only prevent disease, which is the purpose of the vaccines, but to maybe treat infectious and potentially even more importantly, non-communicable diseases, I think, which would be very good for the mRNA field in general. But I think that what we saw today was an unlocking of many of the, or a hint of the unlocking of many of the potentials. But there are still questions. The question around durability, was it antigen specific? Was it a consequence of the fact that we couldn't really measure boosts because wave after wave of COVID infections hit us? So it was difficult to see a naive or simply primed state. We have questions about mucosal immunity and the impact on transmission. We saw data on targeting, I think, which is, again, these are the kinds of things that will make mRNA into a differentiated, more effective technology and a platform that will have multiple different uses. Some side effects, and some side effects that are still relatively unexplained. And of course, no one really mentioned access, but for the International Vaccine Institute, access and equity in this technology is really a critical part of ensuring that the next pandemic doesn't impact Korea, but also doesn't impact the parts of the world where these technologies were not available. So I'm going to answer some questions, and people may or may not agree. So will mRNA replace the older vaccines? I think that the answer is unknown at this point, but I would guess probably not, all of them. There isn't really an interest in the companies to develop new pediatric vaccines for vaccines that we already know are safe and efficacious. Will mRNA vaccines work against bacteria? So a lot of the work that we do, actually maybe a little more than 50% is on bacteria. Will mRNA be effective? I think we don't know right now, and it's possible. I think that people are making, again, progress on the differences between bacterial antigens and viral antigens. So maybe there's a possibility, but we didn't really hear much about it this time, but maybe the next time. Is it a mature technology? And I think I tried to make the case earlier that it is not. And it's important that it's not, because that means that there's still a lot of progress that can be made scientifically in helping us understand better how this technology can be used not only for prevention of infectious diseases, but prevention and treatment of other diseases as well. Will low and middle income countries have a chance at this? And I think we didn't really talk about it, but the WHO has put forward these mRNA manufacturing hubs. IVI is participating with WHO and MOHW, Ministry of Health and Welfare of Korea, in the Global Training Hub in Biomanufacturing, which is the first step in the process of helping other countries in other parts of the world develop regional, if not national, manufacturing of mRNA products. And you saw some of the collaborations that exist there as well. Collaboration potentially with Duke University, the University of Pennsylvania, Dan and his group, all have the potential then to spread this technology to other parts of the world. So I think what was really clear here too is the role that the KDCA is playing in driving a lot of this innovation and pushing it forward. I think that's really important. Government support for this kind of research is critical if we're going to differentiate and make it a much more useful technology. I mean, it was very useful during the pandemic, but we think, I mean, many of us believe that there are other beneficial uses for the mRNA technology, and I think the work that the work that's funded and pushed forward by the Korean government and its agencies has been really critical. In the future, self-amplifying vectors have the potential, again, to be really interesting and important, not only in COVID, but in other diseases as well. The rise of CEPI and the importance that CEPI plays, and actually in many of the companies had mentioned this, and you see the CEPI logo featured, not only for outbreak diseases, I mean the pandemic preparations, but also for diseases that may not become pandemic, but which will certainly be of regional importance in terms of outbreaks. And I'm thinking particularly right now around the chikungunya outbreaks that are happening in the Indian Ocean again. Correlates of protection and mucosal immune responses were brought up. The importance of these, the work that AI and supercomputers and these other things can do in helping us design better immunogens or antigens. And it wasn't really mentioned, but this is a particular branch where the bacterial antigens are different, because in bacteria they fold differently. And the use of AI to try and predict the folding and to try and develop new structures based on that structure, I think, or new sequences based on that structure, could be very important as well. But so again, all things to keep moving forward. New mechanisms to deliver vaccines. So the LNPs, we learned there are a host of new and different ones. The side effect profiles are different. The targeting can be different. I think targeting is maybe one of the next steps, because this, again, gives mRNA an opportunity to get into different cell types. Maybe not important for all the infectious diseases, but certainly may be important for cancers, autoimmune diseases, and other potential targets. Delivery through microarray patches, again, something that we've been very interested in as well. Because from a global health perspective, the patches offer the opportunity to provide vaccination much more widely, much more quickly, and without needles and syringes, and potentially with easier logistics, so heat stability for extended periods of time. The last section, I think, was really interesting, because and so those of you who are not from Korea won't necessarily remember that Green Cross has been around for longest. SK and Eubiologics are newer, and then we saw, SML, QuadMed, other brand new companies, and this is the evolution of biotechnology. And really represents the growth of companies, of commercialization opportunities out of the technology that's presented by RNA. And we've really actually just scratched the surface. I think if you talk to any of the people in the business side who are in the BD side of that, the biotech community in Korea is very active, and companies like Lemonex or ST Farm were not here. But they are very active parts of a very vibrant ecosystem around mRNA. So what will this look like in the future? We don't know. And I think that's, in a way, a good thing, because at the rate that it's growing, there will be progress made. And I think this has been a very hopeful and meeting, and I'm really thankful for all the speakers who came and gave up their time, some of them traveling halfway around the world to come here, and also to the audience who, I mean, we've done now 6 of these, and this is definitely one of the most exciting and best attended of the Global Vaccine Forum. So thank you very much for coming, and thank you again to the speakers.
Thank you, Jerome. To echo your thanks, thank you to everyone for joining us today. This concludes now the 2025 Global Vaccine Forum, and we invite you to the lobby for a cocktail networking reception. Thank you.